the area. So we have to produce more and more from the same given area. That is the reason I have shown that our agricultural resources are limited, but our need is more because our population is increasing. Right now we are 1.41 population. It came from 15 billion to 1.541 over the period, and accordingly we have increased our food production also in phase with the population. So if you see in the food production in 1950s, our food production was only 50 million tons. But see today, we are around 300 million tons. Such a jump has been done over the period. But if you see the area available for cultivation, it's only 140 million hectares, which was available at the period of 1952 till date. That is, there is no scope to increase area of cultivation, but you have to ask the plant to produce more and more using various technologies to ask the plants to produce more and more to meet our need. So this is also an interesting phenomena research where we are making genetic improvement of plants to produce more using various approaches. And if you see the last year, and out of 129 million hectares, we have produced 369 million tons of food production we have done. But if you see the future requirement, it is around 343 to 380 million tons. That means our need is keep on increasing, but accordingly, we have to produce more to meet the increasing demand. So that is the reason that why we are working in DAE for an agriculture, because if you know that this is we have the nuclear agriculture and biotechnology division, and if you say agriculture, these are the components of various agriculture research. If you say research, there is a agriculture, there's agriculture crop plants, rice, wheat, sorghum, something like that, and we need the good soil to support the plant system. And there is an insect we have to control, and there's a disease we have to control, and the nutrient uptake and the availability from the soil, improving the soil carbon, abiotic stresses and heavy metal tolerance, which again from the soil, controlling soil-borne disease. So roughly these are all the, what I can say, the areas which we are working to increase the food production. If you see the pest control, we are using radiation to develop a technique called sterile insect technique to control insect pests. Again, we are using radiation to improve certain fungus which have antifungal activity to control pest and disease. And we have radioisotopes to study this nutrient uptake from soil to crop plants. And we are using different spin-off technologies like biocompost and improved microorganisms to convert agricultural waste into suitable farmyard manure. So those again it is used in the soil to increase the soil carbon. Again, we are using various biostimulants and other approaches like bioremediation to recover the soil from heavy metals. And again, the soil controlling the soil-borne disease with antifungals like trichoderma. So why crop improvement? Among these various research, right now I'll cover only the what research we are doing with the crop improvement, because this crop improvement is a continuous process. You know that all crops evolve from its wild species since human beings are cultivating. So it's a continuous and evolving process. And we are having a lot of current challenges like climate change. You know that there is a increase in temperature and increase in carbon dioxide, increase in unforeseen rains and drought. So we have to improve the plants to adopt all these challenges. That is where our breeding is continuous, and we have to address all the emerging pest and disease and environmental condition. If you see the objectives in the crop improvement, mostly it will be revolving around all these four main objectives. The foremost objective will be increasing the yield, along with quality of the produce what we are eating, like reduced anti-nutritional factors, improved iron and zinc availability, and abiotic stress tolerance, and pest and disease. See, overall, these are the objectives of any crop improvement program which we are dealing. And these are our target crops which we are working on, like cereals, rice, wheat, sorghum, pulses, mung bean, urad bean, pigeon pea, cowpea, chickpea, oilseeds, groundnut, mustard, soybean, linseed, sesame. We do work on vegetatively propagated crops like banana, sugarcane, ornaments, pineapple, turmeric, and ginger. Okay, if you see the breeding strategies, how to improve these crop plants, these are the strategies normally we follow. See, if you say any crop improvement, there should be a genetic variability. That's what we call genetic variation. How this genetic variation comes through different process like mutation, introgression from wild species, and recombination process, tissue culture, and alter by design, what we call as CRISPR-Cas method. All this contributes to increasing the genetic variation. And the other side is to select the better plant from this genetic variation, for which mostly the selection is done on the phenotypic effect. That is what we call morphological selection. But during the different increase in the genomics approach. Now we are following forward and reverse genetic approach to select the plant. That I will be dealing a little bit in my talk, how we are using these approaches in selecting the better plant. So you might have all heard about the green revolution in our country, which happened in 1960s. The wheat varieties, which are, we might have heard about the Norintan and DJ Wujen. These are the rice varieties which created revolution in the country, because in those days our rice and wheat varieties grow very tall, and because they are growing tall and they cannot withstand and they fall down in the windy condition. But these dwarf varieties, which we imported from China and Japan, wheat, this Norintan gene from wheat, and this Diogeo Wujen from this beta rice variety, sorry, this Diogeo Wujen, where semi-dwarf characters are brought from these genes. Fundamentally, if you analyze at genomic level, one base substitution at the RST gene of the wheat has resulted in the semi-dwarf character, and same case in rice, the 383 base deletions as in this particular gene OSGA20X resulted in the dwarfing nature. So basically, the slide says that mutation plays very important role in the our green revolution. That's where I kept this slide. So from that slide, you might understand that the mutation is happening in nature. So we are trying to create such variations using these physical mutagens like X-rays, gamma rays, electron beams, and asteroid particles, as well as chemical mutagens, as well as biological method like DNA insertion, tissue culture. So this, you may be all aware of these mutagens which we are using. And for the benefit of the student friends, normally how we follow this mutation breeding approach, we have a gamma radiated cell where we have a cobalt-60 source, and we irradiate the cell with radiation, and we grow the plants in the field, which we call as M1 generation, and we go to the next generation, what we call M2, and this is how the genetic composition changes from M1 to M2. You can see the recessive mutation here, and it goes to homozygous recessive mutation. Then we can find out the mutant in the M2 population, and in this mutant population, you can identify the what you call the record for our purpose. If it's a large seed size or whatever the trait we are looking for, we can select. This is how we go through this process. And the success depends upon selection of appropriate mutagen, effective doses, screening method, and large population size. Then you may be asking question, why only mutation breeding, not as in many other approach? See, the mutation breeding has a unique approach. It can create new genetic variability in crop plants, and it can improve one or two traits in a well-adapted variety. For example, if there's a very good variety, but it is susceptible to particular disease, that can be improved through this mutation breeding. And to break the tightly linked genes, sometimes when we transfer from genes from wild species, there is always a tight linkage between two triads due to the presence of the genes together, and they cannot removed by recombination. So the radiation-induced changes can only alter such type of tightly linked genes. That is where the radiation plays a vital role. And to improve the vegetatively propagated crops, vegetatively propagated crops like banana and other crops where the recombination is not possible to create genetic variability, the radiation-induced mutation is the only way to improve the vegetatively propagated crops. So this shows that how the radiation plays a key role in genetic improvement of crop plants. So quickly to showcase some of the important traits induced through radiation. So you can see the sunflower. Normally you would have seen 3-4 feet sunflower. You can see how dwarf it can be produced through radiation effect. And here it is a soybean. You can see this is a parent, and you can see the different gradation of height reduction happened due to radiation. And I will not discuss about the economic use of this trait, but just I will show you how these traits are getting changed. But in case of pulses chickpea, you can see the early maturing mutant, and in case of linseed ulci, you can see the this particular line is flowering very early compared to the parent. Again, in mung bean, the early mutant, and in soybean, again early mutant. So I just showing the different early characters in different crops because they have unique application when you consider the farmer's point of view. The farmers can harvest the crop well in advance so that it will not undergo any further stress at the harvest season. And again, this is we can induce disease resistance also through radiation using mutagens. So this is one viral disease, what we call as yellow mosaic viral disease. And you can see how the entire field is infected, and you can see the resistant plant. Not even a single plant has been infected by the virus. That shows how the mutation has happened in a particular gene to protect the crop from the virus. And again, some of more examples for the disease resistance in soybean. Again, you can see the susceptible one. And in groundnut, again, you can see this is a susceptible one, and you can see the good plant standing in the field condition. This is how we select the plants which are resistant to particular disease in a given field condition. And again, in wheat, the fungal diseases are very important in wheat. You can see this is a susceptible one and a very good plant standing in the field alongside, which is also developed through radiation. Okay, so apart from disease also, we can develop some of the unique characters like root length, which will have a unique application in case of drought tolerance, where it can uptake more water from the down deeper layer. So this is a field testing where this type of long root mutants are standing in the field condition where the adjacent plants are already dried. And altering the plant type. See the plant type alteration, for example, here you can see the tall plant and it's a dwarf one. The parents are looking like this small one. Nowadays, we are going for mechanical harvesting, that is cutting the plants with machine. It needs different kind of plant type so that machine can cut here. If you cut here machine, you can see some of the branches may go in the field. So we need to alter the plant type. So again, here in blackgram also we have altered the plant type. See the phenotype of the parent and the mutant. So the machine can normally cut here without any problem. Such type of altering the plant morphology is also possible in mutation. And you see the change in the pod size and the venations of the pod. These are again some of the pod size variations which we can see from the mutant population. Again, this is an example of small seed and large seed which can be developed through radiation. Not only the agricultural crops, we do create variation in flower crops. See, there's a carnation. This is a parent type, and this is the variant. You can see the different kind of thing. And again, in chrysanthemum and other flowers, how the variations are created through radiation. Again, in some other flower crops like gladiolus, you can see the kind of mutations which we are getting the doublet colors. And these are some of the variation we create through mutation. Not only the morphological variation, we do create variation in the biochemical characters. For example, in this crop and blackgram, this is called oligosaccharides. These oligosaccharides are present in the pulses, which creates the blotting. When we take these pulses, we get the blotting. So this is because of this oligosaccharide. And the oligosaccharide is nothing but the polymer of this raffinose, sucrose, webuscose, azygose, and what we call this as RFOs. So because of this RFOs only, we are getting this bloating. So what we do, we did a mutation breeding experiment. We identified a mutant which is having only 50% of the oligosaccharide content. That means that it may have reduced bloating effect, that which have to be studied. And not only the oligosaccharide, now the biofortification is coming up a big way in the country because the iron and zinc requirement for the population is very much required. So through mutation breeding, we are selecting the mutants which can accumulate more iron from the soil. See, the parent is having only 9.15 milligram per 100 gram of iron, but we are having some mutants which can accumulate up to 15 grams. This is an example how the mutation can alter any type of trait in a given plant. So what we are doing with these mutants will go directly to the farmers as a variety if it is having all the unique characters, or we use these mutations in mutant into mutant crossing, or you mutant with cultivar crossing. We do all sorts of combination crossing and follow the progenies to select the better plants, and then we go for evaluation. After evaluation, it will be released as a variety. So through this process, BRC has released 55 crop varieties in different crops. Groundnut 16, this number indicates the different crops and the number of varieties released from each of these crops. So basically the process goes like this. BRC, we develop the mutant, and after stabilizing the mutant, we give it to the Indian Council of Agriculture Research and state agriculture universities for evaluation. And after evaluation for 3 to 4 years, it will be released by Ministry of Agriculture and Farmers Welfare through the Gazette notification. Then it will be go to the BRC as well as state agriculture universities for seed multiplication and further deployment to the farmers. So normally it takes 10 to 12 years for developing one variety. So this year we have almost released 8 crop varieties this year. This is just to see that our achievements are still coming. And this is a recent our mustard variety, Trambacola 1081. So you can see the very high yield potential, and it is having a very large seed size. That is the unique characters of those variety. And this is our new variety, TAG Trambacola groundnut 73, which is having high yield. It is having 23% high yield over the existing variety, and also having very good oil content. Okay, so this is a very good unique example of our mutation breeding achievement. This is a ULSI, what you call linseed. See, normally the linseed oil is not used for consumption because of its poor keeping quality. The normal variety is having 40% linolenic acid. Because of this linolenic acid, the keeping quality will not be there for more time. So people don't use it to be used for industrial purpose. But through our crop improvement program, we have reduced the linolenic acid from 40% to 2 to 5%. Now this crop has become the edible oil category. So that is where ICR has declared the linseed oil to join cooking oil category. This is the first of its kind in the country where we have developed a variety which is from non-oil edible purpose to edible purpose. And again, cowpea, this is what we call chowli. It's a direct mutant of this particular genotype. This is having very high yield and also suitable for summer season in the northern region, and is also resistant to the cowpea mosaic disease, which is a serious disease of the crop. And if you see the rice, India, different parts of India, we have different rice, which is having its own unique character. For example, you can see the Dubraj, Java Food, Vadakolam, and this name indicates that they are popular in different parts of the country, and they have unique characters like fine grain, aromatic, medicinal value, nutri-rich. See, because of these properties, farmers are cultivating bucket, but these rice varieties are not high yielding because they are growing very tall and they're large and they are having poor agronomic character. So now what we are doing is that we are, through mutation breeding, we are trying to improve those traditional varieties which are having unique characters, and we are having a collaboration with Chhattisgarh IGKV Raipur, and this is one example. You see, this is the Dubraj rice normally grown in the field, it will be lodging like this, and through mutation breeding we have developed a dwarf mutant which will not lodge, and now the farmers are growing this particular variety. And also to show how this technique is being used worldwide, if you see the success of the mutation breeding in world, around 3,365 mutant varieties has been registered, and mostly in cereals, followed by flowers and legumes. And different continents, if you say Asia is leading in mutation breeding experiment. And if you see the radiation, this is the idea I want to just to show. The gamma ray has contributed to development of 64% of the varieties, and followed by X-rays. Okay, now we are working on alternate mutagens like electron beam. Why electron beams and other ion beams are required? Because we need better radiobiological effect because of the linear energy transfer. And recently we have done this electron beam experiment with various crops, and we found that electron beam is giving very good frequency of mutations. And this is some of the examples of electron beam induced variations in different crops. Like you can see the high yield, and you can, this is one of the peculiar example. The tall plant you can see on the left side has been reduced to this dwarf type using radiation, which will be again useful for the farmers. And not only crop plants, we are using the fungus, this Trichoderma virens, which is an antifungus kind of antifungus fungus. So you have used radiation to improve this fungus, and we have found that this is giving better efficacy in the field control of controlling the disease. This is the Trichoderma treated field, and this is the non-treated one. So we are also using the genomic approaches like molecular markers for selecting the crop plants. And initially we started with the random markers to develop linkage map, because the plant characters are again monogenic and polygenic traits. So to transfer the polygenic traits, it will be very difficult to follow morphologically because of the environmental influence. So we follow these genetic markers. We developed such type of linkage map to map the genes responsible for monogenic and polygenic traits. So this is one of the example I can show that how the insect resistance has been mapped using this genetic map, so that it can be, see, this is a location of the insect resistance which we are working, especially the storage pest brookit. And so if you follow this marker present in this location, it ensures the transfer of the gene responsible for the polygenic trait. And we have done the transcriptome analysis, and then we have shown these different anti-insecticide properties present in these wild species. I'll just skip some of the slides for want of time. So we are also working on improving the genomic resources for crop improvement. We are doing the whole genome sequencing so that we can develop more markers for crop plant, and we can analyze the mutations. This is one example where we have analyzed the transcriptome level of mutants, like the cowpea mosaic virus and the resistant mutant and transcriptome level, when we have found that some transcript very unique to the mutants, not present in the parent. Such type of information we are collecting. Recently, we have analyzed the gamma ray induced mutants through whole genome analysis, and we have taken 3 mutants for the analysis. This is the virus resistant, large seed size, and small seed size, and we have analyzed the 3 mutants at whole genome level through the NGS, and you can see the indels and the total variants and the inversion to the transition to transversion. Mostly the single substitutions are very high in these gamma ray induced mutants, and these are some of the information which we are trying to extrapolate to understand the nature of mutations in different mutant populations. Now we are going for the speed breeding because as I told that developing crop varieties takes more time. Now we are using such type of greenhouses where we are growing plants in the continuous light so that it can flower early. So that is the idea behind this. And using this approach, we are trying to reduce this period from 3 to 7 years to 1 to 2 years. And so the other parts remain the same. That is what we are doing in the speed breeding. And we are following the MuteMap and MuteMap plus QTL sequence, which are all genomic based selections where we can find out the mutation specific to the trait using the NGS, and so that it can be transferred to other plants. So this is the small way to show how we are following this MuteMap or MuteMap plus procedure to identify the mutations. Once we have the mutant population, we bulk the particular phenotype, DNA of the particular phenotype, and then go for NGS to find out the variants. So we are also working on, just 1 minute. So we are also working on tissue culture to propagate the crop plants, and this also used to in vitro mutagen is to exploit the variations. And these are some of the vegetatively propagated crops where we have developed a tissue culture technique. And also we work on the medicinal plant to compose this anti-cancer compound, where here our idea is to pathway modification so that we can produce more composites through this procedure. And just to show the last, not only we develop, we also deploy these varieties, being in Madhya Pradesh, and this is a TJT501 Tur variety, which is popularly grown in Madhya Pradesh, just for the information. And this variety is very popular in this Madhya Pradesh because this has been grown this Jawaharlal Nehru Krishi Vishwavidyalaya collaboration. You can proudly say that in Madhya Pradesh, whatever the Tur we are eating is from the BRC variety, we can proudly say. And to conclude, our research in our deep agriculture focus from mutant germplasm development of molecular markers, and developing technologies for the plant and soil health, and ultimately leading to societal development. This is how we are working. So acknowledgement goes to Indian Council of Agriculture Research, State Agriculture Universities, Department of Atomic Energy, BRNS, and International Atomic Energy Agency, Vienna. Thank you.
Thank you. Professor Subramaniam, just one question we can have.
Okay, so very good. I mean, you are doing wonderful job for the benefit of farmers, right? So since you are developing the mutations and to make the insect resistance, right, and to improve the, what is your outreach program? How do you reach to the farmers in the interior? Because all these applications belongs to the interior part of the country, not the city mostly, right? So what is the outreach program? How do you find it easier, or my point is, I don't see in, for example, in the north interior that people are aware of these kind of things.
That is the last slide. The very purpose of the last slide is to show that our varieties are very popular in Madhya Pradesh. So normally we collaborate with state agriculture universities. Once we develop the variety, we give to the university. So they will produce and give it to the farmer. They have the mechanism to spread the variety. And we have collaborate with national seed corporations and other NGOs to spread our varieties. We also do the outreach program on our own.
Okay, thank you very much, and many congratulations to you, Dr. Subramaniam, for doing a very nice work.
Now I request Professor Dubey sir to kindly present a memento to Professor Subramanian sir. Thank you, sir.
So we move to next speaker now. The next speaker is Professor Nandini Verma from TMC Attract, Navi Mumbai. Have I spelled the name correct? She, Nandini Verma. Yeah, it's written she, so I'm a little bit confused. Okay. The research interests of Professor Verma include cellular signaling through cell surface receptors, heavy metals, ion redox in drug resistance and EMT, epigenetic regulation of drug resistance and metastasis, tumor secretome-driven targeted therapy. Her scientific recognitions are ACRB, SRG, DST, India, Bushland Fellowship, Bushland Foundation, and McDonald Lipman Grant Israel, Senior Postdoctoral Research Fellowship for Academic Excellence at Finberg Graduate School, Israel, and VATAT BC Indo-Israel Fellowship for Outstanding Postdoctoral Researchers, Israel. So I welcome Professor Verma to give and deliver a talk.
Very good afternoon to all, and thank you, Dr. Dubey, for the nice introduction. So first of all, I wish to thank the organizing committee, especially Dr. Majumdar and RRCAT and HBNI and DAE for this meeting. And this is the very first meeting that we are all together in the life sciences and we can collaborate. Since all biological molecule has to collaborate and the same phenotype we all need to show. So basically, I'm working mostly on triple negative breast cancers, and I'll show you some of the approaches by which we can utilize certain targeted therapy and can reduce the metastasis as well growth of these tumors. So to start with, yeah, so it is needless to say that breast cancer is the topmost cause of death in the females through metastasis. And if we look at the origin of breast cancer, so this is basically can be of luminal origin or it can be of basal origin. And there are basal epithelial and luminal lineages in the breast. And the tumor can originate either from the ducts or from the lobules of the breast tissue. So because breast cancer is a heterogeneous group of disease, and it has been classified into luminal A, luminal B, HER2 positive, and triple negative breast cancer based on the expression of the surface cell receptors. And as you can see, that breast cancer is having the worst prognosis when it is triple negative, which means pathologically the expression of estrogen receptor, progesterone receptor, and HER2 amplification has to be negative in all these triple negative breast cancers, which are the major targets for the therapy of the breast cancer. But on the same time, you can see that Ki67 score, which is the proliferation index marker, is very high in triple negative breast cancer. So that means you can utilize chemotherapy to treat those cancers. However, the outcome of the breast cancer TNBC patients are not so good, mostly because of high proliferation and aggressive behavior of these tumors and very high rate of metastasis that they have based as compared to non-TNBC, which are mostly targeting bone and liver. So another thing that I want to highlight, this metadata analysis as well as several data collected from the clinical research indicate that India has the almost is topping in the list of % prevalence in triple negative cases from the last decade. And we are among Uganda and Pakistan and Ghana, and you can see that in certain East countries also this is high prevalence. So that means there's an utmost need to understand this kind of tumors, especially in our population, and to find out what are the factors that actually control their aggressive behavior, metastasis, and drug resistance. So when a TNBC patients come to the clinic, the they have actually, the treatment regime starts with the chemotherapy, which is neoadjuvant chemotherapy, which is combined with cytotoxic drugs. And after the tumor regress or there's a non-regressive disease, then they can surgically resect the tumor and then further radiation therapy is given in that cases. However, not many patients get the complete therapeutic response with chemotherapy, and even after removal of the tumor, they get recurrence and metastasis, and they die within 5 years of the detection of the disease. There are some therapies which are of targeted therapies, which means where you have to have an expression of certain molecular receptor or a molecular target that is actually controlling the behavior of the cells. And if you target those molecules, there is a high chance that the tumor might not come back or will die very efficiently. However, there are very limited choices of targeted therapy that can be applied to triple negative breast cancers, which are usually PARP inhibitor in case of BRCA mutations, PI3 kinase or mTOR pathway inhibitors when the tumor has loss of PTEN phosphatase, and when you have androgen expression in those receptors, or there are some antibody drug conjugated which are actually targeting polymerases, and there is a fair chance of immunotherapy if the tumor is expressing PD-L1. So why we need to worry about TNBC? This graph can well explain. So if you see the complete pathological response, which is PCR, in TNBC versus non-TNBC patients of 7-year survival, you would see that if the patient is doing good and is treated completely without residual disease, the chance of getting survived for the patient of TNBC versus non-TNBC is not so different. However, when you see that the TNBC patient has a residual disease, they die much more faster as compared to non-TNBC disease carriers. So there's the need of the hour is to increase the number of patients that can achieve complete pathological response and to find out a treatment option for the patient that actually get resistant to chemotherapeutics. So as we know that breast cancer itself is a very heterogeneous group of disease, and TNBC itself is furthermore heterogeneous. So in order to understand that, Lehman et al. Group actually put good efforts to find out molecular subtypes in triple negative breast cancer by mRNA expression data analysis, and they could cluster those TNBC patients into different subtypes by the expression of certain marker genes that are highly expressed in those particular subtypes. And the major subtypes they actually could classify from basal and non-basal to basal 1, basal 2, immunomodulatory, mesenchymal stem-like, and liminal androgen positive. And they were fairly different with having fair difference between their survival and outcome in those patients. So currently we follow the Lehman et al. Classification for the breast cancer based on the molecular subtypes. So as you can see here, the response to different subtypes of chemotherapy, subtypes of TNBC is very different when it comes to chemotherapy. The very good survival rates are found in basal-like 1 patients, followed by mesenchymal patients and mesenchymal stem-like, and liminal androgen and BL2 type of patient actually respond very poorly to the chemotherapeutics. However, the molecular subtyping also find out certain biological pathway which were activated specifically in those subtypes, and you can see there can be a promising range of targeting factors that you can apply in order to understand how we can treat those subtypes differentially and better. So in order to imply that, there are several challenges that we need to understand when we talk about targeted therapy. So most of the time when you combine 2 of the targets or more targets together, there is a fair level of toxicity, and that the 2 targets has to work in a synergistic manner. Only then you can find a therapy that will be better and sustainable. Second difficulty is that there are metastatic diseases which have a very aggressive residual tumors, and they are metastatic. So for those tumors, you need a certain different class of targets that can be combined and can find out and can trace and metastatic cells in the body. And the third thing is that TNBC group itself is a very heterogeneous disease, and for that we need personalized therapy for subtype-specific treatments. So in order to do that, we need to understand few concepts so that we can apply them in designing those kind of therapies. So probably everybody is aware of synthetic lethal effect, which is a genetic concept where we see that there are two genes, A and gene B, which together actually can control the survival or fitness of the cell. However, when one gene, either A or B, is knocked down or taken out, the cell can still survive somehow with a backup pathway. But if you knock down or you take out both function of the gene A and B, the cell cannot survive. So that means A and B gene actually are producing a synthetic lethal effect. So if you suppress both of them together, the cell has no other way out to sustain its survival. So similar effects can be also found in a reversible manner, that you can say that synthetic rescue effects. The synthetic rescue effects can be determined by having a vulnerability factor, which is making a cell vulnerable if you target that. So that means if you target a vulnerability factor, that can be anything, any signaling pathway or certain addictive pathway that the cells has, but you also has a rescue gene. So that means there's a partner that if it is upregulated, it will produce the survival and can bypass the vulnerability factor. In that case, the upregulation of the rescue partner would be necessary, and this is called the rescue upregulation. And in the second case, where you have the same effect but in an opposite manner, where suppression of a vulnerability factor would produce cell death, but to get the survival back, you need to downregulate certain another factor, which is the downregulation of the rescue factor, and then you can get the cell rescue back. So that means there are interaction between genes, pathways, and even networks that can produce certain effects, and we need to understand that effects and can have prioritization of those networks that can be effectively targeting the cancer cells. So in order to do that, yeah. So that means these target interaction can be tumor specific, and then they can be working in a sublethal doses if they are interacting in a synthetic lethal manner or maybe in another way. So that means, so I'll take you towards certain examples that we have worked on these concepts one by one, and then I will also let you know that what we currently are doing in the lab. So for the first example, where we talk about synthetic lethal interaction of the targets, so we found that there's a synthetic lethal targeting between BET inhibitors, which is bromodomain protein that Dr. Chandrima actually told yesterday about BET proteins. So this is a chromatin reader protein. And we discovered that if we combine this BET inhibitor with two different targets, one is proteasomal inhibitor, bortezomib, which is already a clinical drug, and another factor, which is CXCR2 inhibitor, which is IL-8 receptor inhibitor. With those therapies, you can actually produce an effect which is subtype specific. So to start with, this was a result of high throughput screen, where we actually taken 17 different targeted drugs, which are highly expressed in the targets are highly expressed in TNBCs, and we then did the IC50 of all these drugs individually, and then we went back to 1/10 of the concentration of these drugs to produce and combine synthetic effect, because the concept is the two targets has to work in a very synergistic manner. Only then they can produce synthetic effect, and the effect also has to be specific to the cancer, not targeting to the normal cells. So for that, then we combine these single end combinations of these drug targets, and then we waited till 72 hours and did viability assay. So what we had, we actually were interested in that you will produce a less viability in a less doses of combination. And two drugs can work in an either synergistic manner, where you have low CI index, combination index. If the combination index is 1, there's an additive effect, and if you have a CI which is above 1, you have an antagonistic effect. So this screen that we carried out with 13 different cell lines, which represent triple negative breast cancer subtypes, and we combine them all together. So this is the screen result. So you just need to focus on the blue and yellow area. So yellow area means cell death. So you have higher cell death for certain combinations, which we have specifically for the mesenchymal subtypes. So there were several, and there were some which only affected basal 1 and basal 2 subtypes, but there were several who could actually affect all the TNBCs, whichever were tested, but it did not affect the MCF10A cell line, which was actually a normal memory grand cell line. However, we also find certain combinations which are also toxic because not just they targeted TNBC cell line, but also targeted the MCF10A cells. So with one of the collaboration with Dr. Ethan Rupin and NCI, he could predict the synthetic lethality score in patient samples, and we could see that some of the drugs which we had has very high correlation with their in silico data that they have generated independently. So in short, we generated 9 mesenchymal specific combination targets and 14 generic, which all worked on all the TNBCs, and some of the were working on the basal line. So out of these, we selected 2 combinations. One is BET protein BRD4 inhibitor and CXCR2 inhibitor for mesenchymal subtypes, and one BET inhibitor and proteasomal inhibitor altogether for all the TNBC cell lines. And then we titrated those effects in lower concentration, and in some of the cell line of TNBC, we could see that they are working also in the lower concentration. And here, this is a cell viability assay where blue color indicates viable cells. So they were not working on MCF10A as well as on MCF7, which is non-TNBC cell line. Similarly, the other BRD4 and CXCR2 targeting, which is specific for mesenchymal subtype, was specifically targeting the mesenchymal subtype in the titration. More importantly, that the BRD4 and CXCR2 inhibition combination were also also were more specific for the paclitaxel resistant chemotherapy resistant cell lines. So just to introduce, BRD4 proteins, actually bromodomain proteins which bind acetylated histones and can recruit all the machinery for transcription and translation. And then there are inhibitors which actually are having anti-cancer properties as well as anti-inflammatory properties. What they do, these inhibitors actually bind to the bromodomain pockets of the BRD4 and will not allow to bind it to the chromatin, so that the transcription will be stopped. So these inhibitors are actually already in clinic, and what we show here that all these cell lines, which is breast cancer cell line, align on basis of their mRNA expression of BRD4, and you can appreciate the BRD4 expression is especially high in triple negative breast cancer cell lines, which is not there in the any other subtype. And then we also looked at the patient cancer database from TCGA, and we can see that basal-like tumors, which are mostly TNBCs, have the highest expression of BRD4 protein. Then we also looked in some tissues of TNBC versus non-TNBC breast cancers, and we could also see a very nice nuclear localization of the BRD protein, which is highly expressed in the TNBC specifically. So there are certain studies that certain groups like Carolina Pollier group, they have already established that BRD4 proteins actually are very much targeting triple negative breast cancer cell line as compared to non-TNBC. They could use several BRD4 anti drugs. And then there are also studies that phospho-BRD4 protein actually is a marker of bad prognosis, and especially in TNBC. So that means it makes a valid target, and we are looking at the correct target to combine with. And especially there are oral drugs which are already approved in the clinic for BRD4. So then we went ahead and tested this combinations in mice, and we could see that the combination of OTX-015, which is a BRD4 oral inhibitor, with SB22522, which is a CXCR2 inhibitor, could also suppress the tumor mice growth of the mesenchymal MJ231 cell line. And also the other pan-TNBC combination, which is the bortezomib and BRD4 inhibitor, actually could also suppress a 41 mice model, which is a syngeneic mice model for TNBC. Then after we also checked the effect on the toxicity of this combination, and it was not toxic to the mice. And even after removing the drug, the tumor could not grow back. And there was a fair advantage of survival in those mice who were treated with this combination. So then we asked, what is the mechanism of this drug? So then we went ahead and checked certain cell death pathways. So for CXCR2 combination with BRD4, we could immediately see that there's an induction of apoptosis, which is an annexin V, which is membrane, we can recognize membrane flipping, which is a classical behavior of cell membranes when the cells undergo apoptosis. And there was a caspase activation as well as FAST3 was also seen in this combination, which is specific to mesenchymal cell lines. However, if the other pan-cancer combination with BRD4 with proteasomal, we could not detect apoptosis, we could not detect also necrosis, and also not autophagy. But however, we know that in that case, there was a high ROS induction in the cell when you combine this targeted therapy to triple negative breast cancer. So at that time, we know that there's another pathway going on, which is working on the lipid peroxidation. And we saw that there is in this pathway, you have a central molecule called GPX4. It's a glutathione synthase molecule that converts the oxidized form of glutathione to reduced form of glutathione, as well as certain pathways are also there, which makes basically the anti-ROS molecule, which is glutathione. And there are certain inhibitors of this pathway by which we can study the lipid peroxidation and ROS. Importantly, iron is also involved by Fenton reaction. And there are certain receptors which bring iron in and iron out of the cell. And by this oxidized form of iron, there's with the help of low glutathione, which cannot repair the enzyme, now they have the ferroptosis cell damage and the cell cannot survive. So this is another cell pathway, that pathway that has come up. And by using inhibitors like erastine and iron chelators and ferrostatin, which is an inhibitor, we can study this pathway. So then we put some rescue experiments. Okay. And then we can analyze that, yes, this pathway is actually ferroptosis. And the specific lipid peroxidation was also seen. We could see that iron intracellular levels were high with this combination and glutathione was low. And then we could conclude that there were some factors like GPX4 was suppressed and NRF2 factor was increased. Decrease in this pathway, same GPX4 was down. So all this pathway indicate that there is a regulation of this pathway in this combination of drugs. So what we see that pan cancer effect was actually with ferroptosis. So we asked the question if TNBCs are also susceptible for ferroptosis by themselves. So we took two inhibitors. One is GPX4 inhibitor itself, and another is erastine, and we could actually mimic the same effect in all the TNBC cell line versus non-TNBC cell lines. The TNBC cells are fairly susceptible to ferroptosis induction. And then we asked why it is so. So we found a very nice explanation for at molecular level that the pathway was severely altered with in this way. So this effect was also found in tissues. And then we did some clustering analysis taking 700 genes of ferroptosis, glutathione, and iron metabolism pathway. And you could see a nice clustering in basal-like versus all the rest of the breast cancer. And this is a severe metabolic phenotype that we could see that in TNBC cells, TNBC tumors are fairly unique. So also the ferroptosis pathway was actually enriched for basal-like tumors in the cell and also in the patient level. So when we look at the KET pathway, there were several molecules which are highly expressed or lowly expressed when it comes to ferroptosis pathway, 40 genes. So this is just an example how we actually take the fold change of TNBC versus non-TNBC or gene expression of all the KET pathway of ferroptosis. Thereupon, there's one study of with EGFR where we show that synthetic rescuer was involved. So what we did actually, EGFR is a target in triple negative breast cancer. It is overexpressed in various TNBC cell lines. But if you target it by itself, there's a resistance due to HER3 upregulation, which is another kinase. So what we target when with PIK2, which is an FAK family cytosolic kinase, you can generate a synthetic, this is not there, it is suppressed, the synthetic rescue is suppressed, and you see a fair effect with the combination. So this effect is bring about by NDRG protein, which actually facilitated the degradation of the HER2 receptor by itself in a PIK2 knockdown condition. So this was one of the examples. Another example I will just quickly go through. So this is the mechanism how it does. So with EGFR, you have anti-EGFR therapy actually generates upregulation of the synthetic rescuer, and when you combine with PIK2, this HER3 was eliminated, and the combination actually could provide upregulation of NDRG1 and suppression of several pathways which actually generated the effect and the combination therapeutic effect in triple negative breast cancer. Another, we did some screen with the BET inhibitor protein with secretome, and the idea was if you put IC50 of BET inhibitors, you will get IC50 if you do not treat with any of the secreted factor library, and if you treat them with the library, pre-treatment of the library, and then provide the IC50 values of BET inhibitor, and if you see increase in increase in the IC50, that means there are synthetic rescue effect, and if you see decrease in the number of cells after treatment of the library component, you have the synthetic rescue which is working on a down regulation manner. So we did this screen and we found several compounds which were actually working in this kind of behavior. So we are taking it forward for our analysis for validation of targets, synthetic rescue effect, and so on. So I think I should not conclude here. Probably it's good. Uh, so, so currently what we are doing, we are mostly focusing on triple negative breast cancer in terms of chemotherapy outcomes. So what happens when a tumor is treated with drug, and if you have a residual disease, this can actually all the time can metastasize and can reform the tumor again. And what happens here, mutations are mostly not found in TNBC when it comes to drug resistance, but there's a severe level of reprogramming here. So we need to understand whatever is going on here, because if we miss this thing, there's no chance to treat this tumor when it is metastatic already. So we are trying to understand molecular reprogramming, autocrine and paracrine secretome, and epigenetic reprogramming, because this is most of the time happening in the TNBC patients. So in order to do that, we are building some model which is called drug tolerant persister model, and we are utilizing the cellular clinical and clinical parameters, and we already attained some the drug resistant persister cell on BL1, BL2, mesenchymal, and large subtypes. So we have already calibrated all. We have already did some assays, and we see that these are aggressive behavioral drug tolerant cells that have high tolerance, high tumorigenic potential, high EMT, and ROS and glutathione levels are altered. Many cells by cellular organelles are altered. They have high intracellular iron contents. They have high migration and invasion. And now what we are trying to do some multi-omic approaches in order to understand what are the things that actually make them persistent and how we can try to kill them.
Sorry, time is up.
Okay. So here we want to collaborate so that we can do some metabolic analysis, iron lipid content, and peroxidation analysis, and secretome analysis. So thank you. This is the acknowledgement for my postdoctoral group, which actually contributed to first half of the work, and some collaborator which was on the work. And this is Dr. Mohit Kumar Jolly and Dr. Sumendu from India, which is helping us to find out things. This is my current group. And most of the people are master except Shagufa, she is a PhD student. And I thank Dr. Sudeep Gupta, Director Extract, Dr. Prasanna, DD Extract, and my lab members, and it's HBNI and DAE force. And there's a support from DAE, Indo-Friends Group, and SIR. Thank you.
Thank you, Dr. Nalini Verma. That's a very systematically done and a very elaborate work that you have done. And it's very interesting to know about your work. So we can have only 2 questions.
Sure.
Yeah, Dr. Verma.
Yeah.
You mentioned about your BRD4 inhibitors, and they might be working through ferroptosis processes. Now, as we might know that the in TNBC patients, most of them after having a NAC therapy or a chemotherapy, they suffer from acute anemia and blood loss. So that means that in their probably the iron content or the hemoglobin content will be low. So in that case, what is your treatment, how that outcome you would think?
Actually, this is 2 different things, anemia and intracellular iron. So there is a separate uptake mechanism for cancer cell that they work on level of receptors. So they intake from the blood itself, of course, but I think BET inhibitors are already safely tried. So they are not kind of producing too much deleterious effect for the patients. So in that case, they are quite safe. So anemia and blood loss, and those are the effect of chemotherapies. So if you combine certain therapies like this with chemotherapy, so maybe even for the post-treated patients who are already getting resistance, this therapy might help. And if we try to combine with some chemotherapeutic therapies, maybe we can lower down the doses. So in that case, probably we can help. But definitely the loss of blood cells and hemoglobin is an independent thing, because it is an effect of chemotherapy. But iron intake of the cell is a different kind of mechanism. Thank you.
Okay, thank you very much. Many congratulations to you, Dr. Verma.
So now I request Professor Dubey sir to kindly present a memento to Professor Nandini ma'am. Thank you, sir.
So we move to the third talk of the last session. This is delivered by Dr. Saeed Hassan. He's from Matrek, Mumbai. His research interests include functional and molecular aspects of novel agents in leukemia, understanding the signaling between BCL2 family proteins and CDMs. His scientific recognitions are pre-doctoral fellowship at University of Rome for Tor Italy, European Hematology Partner Fellowship Netherlands, Ramalingam Swami grant by DVT Government of India, and Associate Editor at Indian Journal of Hematology and Blood Transfusion. I welcome Professor Hassan to deliver the talk.
So, good afternoon, everyone. Okay. So, thank you, Chairperson, Professor Mazumdar, and the entire RRK team to put up such an excellent program on such a short notice. And we are really enjoying our time here. So, Professor Eni Hassan started the, this meeting, and now my job is to finish this. And we share the same last name, okay? And this is the only similarity that we have. Okay, so what I'm going to talk about today is targeted therapy in acute myeloid leukemia. It's a very niche area. It's a very specific topic. When I, started preparing for this meeting, so I was thinking, what should we discuss in this meeting? Whether we should discuss the data from the lab and then, or the general idea about the leukemia. But then I thought, let us have a mix of things together. So before I start with therapy-related acute myeloid leukemia, targeted therapy in acute myeloid leukemia, I would like to introduce what is AML. So those who are new and students who are first time, working or listening to this subject. So acute myeloid leukemia is a type of a blood cancer where there is a clonal expansion of myeloid blast in the bone marrow and the peripheral blood. So what happened in the hematopoietic stem cell in the bone marrow, when it goes through various stages of differentiation and maturation, it forms different blood cells. And arrest of differentiation and at any stage, and especially in AML and the myeloid progenitor stage, leads to the accumulation of these undifferentiated cells into the bone marrow, and these bone marrow accumulated cells comes into the peripheral blood, and these undifferentiated cells are known as blast. So if you look at the survival of AML, acute myeloid leukemia, survival is very poor. So if you look at the data from NCI, so the survival, complete survival, patient who are less than 65 years, survival is approximately 35%, and the elderly patient, the survival is only 5%. So very poor survival, and in fact the data from our center, TMC, when we prospectively recruited 149 patients, the overall survival was 37, which is similar to what is reported in the West. The only major difference between the population of the two West versus India, that our population is very young. When they present with the diagnosis of AML, they are around 25 to 30 years, while in the West, the median age at the time of the diagnosis around 55 to 60 years. So now when it comes to the targeted treatment, there are two paradigms in oncology. One is chemotherapy and one is targeted therapy. So chemotherapy, as you know, that you kill all the cells. You don't differentiate between normal versus cancer cell. While in the targeted treatment, you use the specific molecular features of the cancer cell, and these molecular features could be mutations or dysregulated pathways which are specifically associated to cancer cells. Then you target those cells in the targeted treatment. So, this is the example. This is one of the best example in the translational medicine of the targeted treatment. So leukemia, being a blood cancer, is by definition is a metastatic cancer. So blood is flowing everywhere, and so once it starts with metastasis. But this disease, which is a subtype of AML, is characterized by a reciprocal translocation between chromosome 15 and chromosome 17, resulting in a oncoprotein PML-RARA. So this PML-RARA carrying this acute promyelocytic leukemia, it was initially, when it was 1973, it was treated with anthracycline. When Janet Rolle discovered this translocation, so then there was a drug called ATRA, all trans-retinoic acid, which is a derivative of vitamin A. So this ATRA converts the immature blast, which I had shown in the earlier slides, mature into the normal blood cells. So this, discovery of ATRA in the treatment of acute promyelocytic leukemia has changed the paradigm. How do we treat cancer with a targeted treatment, not with chemotherapy? And if you look at the survival, when this chemotherapy, daunorubicin plus cytarabine, was given, the survival of this disease were very poor, hardly 2 years. But now, in fact, this is an old paper of 97, but in 2022, this disease is virtually curable by the two drugs, arsenic trioxide and all-trans retinoic acid. And both are small molecule inhibitors. There is no side effect. So this is an example, how these drugs, targeted drugs, can be used in another treatment also without having any side effect. When it comes to the treatment of AML, for the past 40 years, still, the induction chemotherapy, which is a cytotoxic therapy, 3 days of daunorubicin and 7 days of cytarabine is given. But, patient achieve initial remission, but within a year, most of the patient disease comes back. And apart from that, there are 20 to 30% of patients who do not respond to even this cytotoxic therapy. So this cytotoxic therapy is leading nowhere, but still is in clinical use. However, in the past 2 years, there is an increasing optimism because of the targeted treatment, such as FLT3-ITD inhibitors, IDH inhibitors, anti-CD33 antibody conjugate, gemtuzumab, or BCL2 inhibitors such as venetoclax. So in AML or in general, the targeted molecules can be divided into 3 categories. One, regulator of apoptosis, such as venetoclax, which is a BCL2 inhibitor, MDM2 inhibitor, epigenetic modifiers such as IDH inhibitors, BET inhibitors, or oncogenic kinases and cell cycle inhibitors such as FLT3 inhibitor, pan-CDK inhibitor, or CDK7. So today's discussion, today's talk, I will discuss some of the data that we have generated using this compound CDK7 inhibitor together with venetoclax. So venetoclax actually is becoming a sort of a magic bullet in the treatment, not only for leukemias, even for solid tumors. This is the only drug which are the targeted agents which can be given as a frontline treatment, meaning this drug so far is able to replace the cytotoxic chemotherapy in AML. Rest of the drugs, for example, IDH inhibitor or FLT3 inhibitor, they are again very useful, but they serve only those patients who carry these particular mutations. So these mutations are present, for example, 10 to 15% of AML patients. So a subset of AML patient will respond to this inhibitor, but the drug like venetoclax, which does not distinguish whether mutation is present or not present, so it can be given to all patients. There is no prerequisite of having any sort of molecular aberration that needs to be identified. And how? So what it does, this diagram shows the mechanism of action of venetoclax. In a cancer cell, the pro-apoptotic proteins are captured by BCL2, which is an anti-apoptotic protein. What venetoclax does, which is shown here as a red molecule, it removes the pro-apoptotic proteins from the anti-apoptotic proteins and induces the mitochondrial mediated apoptosis to kill the cancer cell. But even though it's a very successful drug, but still there is a concern because of the resistance is also emerging in the setting. What happens when you block a BCL2, which is anti-apoptotic protein, another anti-apoptotic protein gets upregulated, which is MCL1, to compensate the normal physiological function of a cell. So what happened? So what happened then when this is the venetoclax, when it removes the pro-apoptotic protein and it induces the apoptosis, but in the resistant cells, this pro-apoptotic protein is captured by MCL1. So now apoptosis is again inhibited. And there is a study which has shown that MCL1 inhibitors actually reduces the leukemic burden in human AML xenografts. So what we did in 2017, we entered in a collaboration with Origin, who has developed a compound, CDK7 inhibitor. It was a covalent, irreversible, and orally bioavailable compound. So you'll ask why CDK7? The first question anybody asks. So as we know, the CDKs play a key role in regulating the cell cycle, which is almost universally altered in all type of cancer, and it has a dual role. So CDK7 on one side, as a part of the cyclin H and MET1 complex, it phosphorylates the various CDKs of the cell cycle checkpoint, CDK2, 4, 6, so that cell cycle progress from one stage to another stage. On the other side, what it does, it phosphorylates the RNA carboxy terminal domain of RNA polymerase 2 at serine 5 and serine 7 to initiate the transcription. So RNA polymerase, which is sitting on the promoter region of various genes to initiate the transcription, where the CDK7 required to phosphorylate the carboxy terminal domain, and it also phosphorylates CDK9, which in turn phosphorylate the serine 2 of the RNA polymerase 2, so to elongate the for the transcription elongation. The first CDK7 inhibitor, the covalent inhibitor, was published in 2014. That was TSZ1, and therefore a lot of publications have come, various solid tumors, which has shown the effectiveness of this CDK7 inhibition to stop the proliferation of the cancer cells. And since the idea behind using this CDK7 and the development of this prototype was not to show any clinical compound, it was how, because CDK7 being such an important protein, if you are going to block it, you are not basically blocking, you are reducing the its ability to phosphorylate the carboxy terminal domain of RNA pol 2, but still, there was a lot of concern when it was started. So it controls because the oncogenes normally, they have sort of a transcriptional addiction. So to bring out that sort of transcription in those settings, and if you see here, what CDK7 does, it reduces the phosphorylation of RNA pol 2 carboxy terminal domain, thereby reducing the levels of MYC, RUNX1, MCL1 sort of genes. So many things have appeared after that, but this compound was non-clinical compound. So therefore, Origin has developed compound which was clinical and orally bioavailable. So what we did with that compound, the first thing we look at the carboxy terminal domain phosphorylation status, and in the two cell lines of AML, we found the serine 2, serine 5, serine 7 was getting down-regulated in time and dose-dependent manner. These are the different concentration of XL102, which is a CDK7 inhibitor. So it is decreasing the phosphorylation level in AML. Then we next check how much CDK7 is engaged with the XL102, the compound, whether it is actually engaging. So what we did, we incubated the cells with treated the cells for 3 hours with XL102, then incubated with biotininated TSZ1, and in the cool down assay, what we found, when there was no XL102, so this compound, which is a non-covalent inhibitor of CDK7, was able to cool down the CDK7. So we could see the band of CDK7 in the cool down, but when you increase the concentration of XL102, the occupancy decreases. It means this XL102 is now occupying the site. When these cells were washed to remove the unbound XL102, the CDK7 starts reappearing. So after 24 hours, because the new cells are synthesized, which are making the new protein. Then we check the efficacy of this compound in various AML cell lines. So what we did, the cell titer glow assay, and we used the 6 different concentrations starting from 10 micromolar using the 3-fold serial dilutions. And we found in the different AML cell lines, 6 different AML cell lines, showing the IC50 in the range of 0.1 to 0.2. So it means because these cell lines are derived from the different patients, someone carries FLT3 mutation, someone carries NPM1 mutation, and in fact the HL60, which is cytarabine resistant and sensitive, both the cell lines, the effectivity of this compound was 0.2 micromolar, which shows that it is working without considering any genetic alteration present in the cell. Then we got some excellent model of venetoclax sensitive and resistant cells from MD Anderson Center. So in the venetoclax sensitive cells, the sensitivity IC50 of venetoclax was 0.005, while the resistant cells were truly resistant. And in this setting, when we use this model, venetoclax resistant, and did the combination study using the XL102 of origin with venetoclax, we found even at a very low concentration, we found a very synergistic effect with venetoclax. Then we further collected, because cell line data from the sensitive and resistant cells was very promising. Then what we did, we collected the patient samples directly from the patient. We isolated, we enriched the blast from 42 chemosensitive cases and 12 chemo-resistant cases. And then we treated these blasts, which are isolated from the patient, from 6 different concentrations like we did for the cell line. And what we found, the mean IC50 of derived from the 42 cases was 0.3 micromolar. In the chemo-resistance, it was 0.4, very narrow. I mean, there was no difference, I would say. So it means the resistant cases, which are very a problem, not in India, but worldwide to treat, this is showing an excellent effect. And when we compare this data with healthy PBMCs or healthy normal CD34, we could collect 3 cases from our bone marrow transplant unit. And we again found that CDK7 inhibitor XL102 was working to eliminate leukemia cells and was having very little effect on the healthy cells. So then we look, because we know the CDK7 affect the cell cycle and apoptosis, as I mentioned in the previous slide. We checked after treatment with the cells with the XL102, the cells were arrested in the G1 phase at different concentration, and the phospho-CDK1 and phospho-CDK2 levels were also getting downregulated after the treatment with XL102 at 6 and 24 hours. And then we checked the expression levels of anti and pro-apoptotic proteins after the treatment in the same cell lines. So they were also, anti-apoptotic protein was going down, pro-apoptotic proteins were getting up. So what this suggests, cells are arresting in G1 phase and undergoing apoptosis through BCL1 and downregulation of BCL1 and upregulation of cleaved PARP and cleaved caspase 3. Then in the next, what we checked, the other cell cycle proteins, what was the status after the treatment of XL102. So we found the upregulation of P53, MYC downregulation, and upregulation of P21, which is a direct target of MYC21. So the story goes in that way. Then when you give the XL102, it induces the DNA damage response. In that response, P53 gets upregulated, which downregulates MYC, and MYC, the direct target of P21, is getting upregulated. So all data from the cell line looks perfectly fine. That pharmacological inhibition of XL102 is giving great results, and it looks like it's working through P53 c-MYC axis. So what Tarang is a PhD student from my lab, she has decided to do the genetic ablation of CDK7 to knock out, to mimic pharmacological inhibition of the CDK7 inhibition in the AML cells. So what she found in the knockout clone of CDK7, not treated with XL102, because this is a pharmacological mimic of CDK7. So all well, serine 2 was going down in the knockout clone, serine 5 was also going down. MYC, to our surprise, I'll discuss this, P53 was upregulated, P21 is upregulated, but to our surprise, MYC was not downregulated. MYC being a oncogene, we were expecting in the CDK7 knockout clone, it should also go down like we found in the patient sample, in the cell line data. Then with what we noticed, but the most important part, the gamma H2X, the DNA damage response in the knockout clone was low. And there was a recent paper which says that to bring down the levels of MYC, the DNA damage response is required through P53 axis, but it's not only the DNA damage response, it's the extent of the DNA damage response. So if XL102 inducing DNA damage more effectively, then MYC expression is going down, but genetic ablation was not able to induce the DNA damage so effectively. So probably that is one of the reasons. However, we are still trying to resolve this puzzle. But based on this paper, we are so far convinced that it is because of the level of DNA damage that we found in the knockout clones. In the knockout clones, again, the cells were getting arrested in the G1 phase of the cell cycle. So what we did next, the subcutaneous model of AML. So we treated the animals, we developed AML xenograft, subcutaneous, and we treated the animals for 3 weeks, and then allowed animals to stay without treatment for 2 weeks. So here you can see the vehicle control. 2 doses of XL102 were used, 30 milligram per kg and 60 milligram per kg, and this is the body weight changes. So in the 3 arms, there was no significant body weight changes, meaning this treatment is well tolerated. While when we look at the tumor reduction, so oral administration resulted in partial regression in the 30 mg per kg, while the complete tumor regression at 60 milligram per kg. So this is the control, which is the tumor is keep on increasing, while the 30 and 60 both are at the bottom, but 60 was able to completely eliminate the tumor as compared to the 30 milligram per kg. And what we noticed after 3 weeks of treatment, when we allowed the tumor to grow, so we found in the 30 milligram per kg arm, tumor was coming again, while then 60 milligram per kg, it was not coming again. So it means 60 milligram per kg dose was good enough for these animals. So since leukemia is a disease of systemic circulation, so we developed the orthotopic AML xenograft, and we found the same results using the 60 milligram kg of 60 milligram kg of XL102. We found a significant reduction as compared to the standard of care cytarabine in the XL102 spleen size. And then when we collect the bone marrow of these xenografts, we found MCL1, BCL2, all those protein, all those data that we obtained from the cell line and patient sample were in complete agreement what we found in the animal data. So I'll just quickly, I don't need to show the entire things. Then to find out what are the transcriptional target of XL102, we did a RNA sequencing study. So we treated the venetoclax sensitive and resistant cells to identify the different targets. So I'll not go into the data. These targets were verified by real-time PCR and Western blot analysis, and we found that XL102 shows synergy with venetoclax in inducing apoptosis. I'm not going into detail, but you can ask me question after my talk or after this session. So I'll conclude. So co-targeting of BC, the idea of this project, the targeting one pathway of BCL2 may not be sufficient. So because AML is a very heterogeneous disease where there are dysregulated pathways or transcription addiction. So dysregulated pathways can be targeted by BCL2 inhibitor, which is venetoclax, while the this transcription addiction of oncogenes can be targeted by XL102 CDK7. So this work will be presented by my PhD student Tarang next week in Croatia. So she got a travel grant and a flash talk for this work. And finally, so I have 3 PhD students. Tarang is my first PhD student who did all this work. Deepshika is also here. She has presented work on autophagy in acute promyelocytic leukemia. Akash has joined my laboratory last year. And this is our clinical team. And obviously, Origin being the major contributor through this XL102. And the collaborator who has provided some important model system of venetoclax sensitive and cytarabine sensitive and resistant cell lines. So this is my last slide. Thank you so much.
Thank you, Dr. Hassan. We can have a few questions.
Yeah, Professor Hassan, I have a couple of questions.
Yeah.
See these small molecular inhibitors, this XL102, and this is a targeted molecule in the sense it selectively targets one pathway. That is correct.
Sorry.
But how do you ensure that, or how it is ensured that these molecules are selectively localized to the tumor? I mean, in the xenograft model, I'm talking.
How this molecule is
Selectively localized to the
tumor. So see, blood cancer anyway is not a localized
no,
cancer.
that is fine.
Right?
In blood cancer, I can understand. But in this tumor
Correct.
model.
But for other cancer, for other solid tumors, because this compound basically does not inhibit the CDK7 function. It reduces its ability to phosphorylate the downstream targets. Okay, so downstream targets, for example, MYC, MCL1, these are the genes which are traditionally highly dysregulated in different cancer solid tumors. So if expression is going down, so it cannot work as a single compound. Then it is bringing down the transcription. Then other targeted treatment, for example, in AML, it's venetoclax. For other solid tumors, that could be another agent which can be combined with XL102. So because like if you remember yesterday's talk of Dr. Subramaniam, so industry developed molecule with a very defined objective. Okay, it's the scientist's job, then further academia job to find out, to make a fine tuning and to align it with the different diseases also. So it's not they were focused, for example, AML, right? But I can think of in AML is venetoclax is perfect combination, but in breast cancer that could be another drug which can be a good combination for XL102.
See, another question is this combination therapy when we are talking of. So when there is sublethal dose, and some of the cells will be receiving sublethal response. Is there any chance of further resistance development in those cells which are not killed for say due to this combination therapy treatment?
In the resistant cells?
Not in the resistance development, chances of resistance development against this combination therapy.
So okay, this is nobody can say anything about these things because this is how the drug development happens. So I cannot say at this moment. Currently the data seems very promising, and this is the way forward. But I cannot predict at this moment that there will be no resistance to XL102 in future. I don't know. I have no answer to this question.
Okay, thank you.
Any further question?
I think there is.
Okay, so if there are no questions, I have one curiosity, Dr. Hassan, if you can answer. Particularly the dose that you have given to animal, 60 milligram per kg, that looks a bit higher side. So what is the reason for that? And second, whether the this drug which were used, what is the, is it hydrophobic or hydrophilic? Maybe that is affecting the bioavailability of the drug.
Yeah, so I think that is a question related to the structure of the compound. How TSZ1 is converted to the to make it more orally bioavailable, right? So I have not shown that data because still the things are in under publication. But there were some changes uh, in the ATP binding domain of original compound TSZ1. Okay, with some hydrophobic changes were introduced outside the cysteine 312 amino acids to make it. There were some structural guided design that have done, so that make them hydrophobic. I have the data, but I cannot reveal at this moment. This is one point, and the second point you said is a high dose. You were saying what was your second question?
60 milligram per kg body weight for the animal. It looks to me a bit higher side.
No, it is not. It is not. If we convert this dose in the human dose, so it is not, because when we check the data from the primary cells and the cell line on animal data, the IC50 finally, which will determine how much drugs should be given to the patient. But 30 and 60 milligram certainly is not a very high dose in my opinion. But I don't know how to do this calculation to convert this dose into the human dose.
Okay, thank you. So we congratulate Dr. Hassan for very systematically done work and very elaborate work done, and that's a very novel approach you have taken. So many congratulations to you.
Thank you.
I request Professor Dubey sir to kindly present a memento to Professor Hassan sir. Thank you, sir. Now I request Dr. Sunil Ghosh, sir, to kindly come onto the stage and present a memento to Professor Alok Dubey, the session chair. Thank you, sir. Now let us take a tea break, and after that we'll all assemble here around 4:40 for the valedictory session. I request everybody to assemble back here at 4:30. So here we are, moving towards the end of this theme meeting which started on 7th of September. Good evening everyone. I welcome each one of you to the valedictory function of first HBNI theme meeting on life sciences. Now I would like to request the chief guest of valedictory function, Professor P.R. Vasudev Rao, Honorable Vice Chancellor of Homi Bhabha National Institute and former Director of the Indira Gandhi Center for Atomic Research, Kalpakkam, onto the dais. I request our Honorable Director, Sir, Dr. Shankar V. Nakhe, to please escort him onto the dais. Welcome, sir. I further request Dr. Sunil Ghosh, Associate Dean Life Sciences, HBNI, to please come onto the dais. And further, I request Dr. S.R. Mishra, Dean Academics, HBNI, RRCAT, Dr. C.P. Paul, Dean Students Affairs, HBNI, RRCAT, Dr. S.K. Majumdar, and Dr. Ajit Upadhyay to please accompany him onto the dais. Thank you, sir. Now, I request our Honorable Director, Dr. Shankar V. Nakhe, to please welcome Professor P.R. Vasudev Rao with a plant sapling. Thank you, sir. Now, I would like to request Dr. Shobhan Majumdar, sir, convener of the HBNI theme meeting on life sciences, for giving a brief account of the meeting.
Good evening, everybody. Honorable Vice Chancellor and today's chief guest, Professor Vasudev Rao, Honorable Director of RRCAT, Dr. Nakhe, Dr. Sunil Ghosh, Associate Dean Life Sciences, HBNI, Dr. Sataram Mishra, Dean HBNI at RRCAT, Dr. C.P. Paul, Dean Student Affairs of HBNI at RRCAT, Dr. Ajit Upadhyay, Head of HBNI Activities at RRCAT, Professor H. Pal, Associate Dean of HBNI, Professor Hema Rajaram, Dean Life Sciences, HBNI at RRCAT, Professor Prasanna, Chairman of Board of Studies, Life Sciences, HBN, and then my senior colleagues here, distinguished faculties of HBNI from different constituent institutes, my dear students from HBNI, my colleagues here and friends. So today is the concluding day of the HBNI, first HBNI theme meeting on life sciences, which started on 7th of September here. And this meeting has been held in RRCAT and this convention center, which is one of the finest convention centers in Department of Atomic Energy. And the objective of this theme meeting, as I also told in the inaugural ceremony, that to lay a platform of interaction among HBNI faculties and students across the constituent institutes and OCCs. More than that, you know that this is, as I told you, that this is the objective of all the conferences which are held normally. But the difference, there is a difference here, The difference is that in all the constituent institutes of HBNI, which happen to be the DA units as well, they do not know what are the facilities existing in each of the units. So this is the theme meeting which has addressed to that. And I'm sure that all the participants who have attended and who have listened through all the talks and seen the posters, they will agree with what I say. It is a fact that we didn't know earlier that so much of life sciences are being done, and many a time there are unique activities in each center. So I would not say not everybody, majority are not aware of them. So for the first time they are being exposed to these kind of activities. And there were this symposium was primarily meant for students, HBNI PhD students. And there were 48 poster presentations. And there were 22 talks from different constituent institutes. Apart from that, there was a keynote address by a very eminent scholar, Professor Annie J. Hassan of Kamineni Hospitals, Hyderabad. She is a doyen in the field of genetics, and she talked about genetics and as well as genetic counseling, which is a urgent requirement of today's time. Our chief guest of HBNI theme meeting was Professor Amlan J. Pal, Director of UGC DAE Consortium of Scientific Research, Indore. And there were 2 evening lectures as well. One by first day lecture was Professor M.B. Bera from Longwal Institute of Science and Engineering, Longwal, who is an expert in food technology. And the second day evening lecture, yesterday's evening lecture was by Professor H.S. Subramania, who is the Director of Institute of Bioinformatics and Applied Biotechnology, Bangalore, and also Biocon Chair. So he talked about, because he had a vast experience of industry as well as academia. So these 2 evening lectures were really, really illuminating. And it was felt that more such meetings should be arranged, not only in life sciences, but also in other disciplines of sciences like physical sciences, chemical science, and engineering sciences. And we at RRCAT will be eager to host this kind of theme meeting in future also, both in life sciences and engineering sciences and physical sciences. We do not have chemical sciences that much here. And I would like to thank all the participants, particularly those outstation participants who have come all the way to RRCAT. For example, NICER. There are no direct trains available for the students also, but they have taken the trouble to really come to Indore. And from other places also, like now Chennai also doesn't have direct train connection here, but despite those problems or constraints, they have come here to make it a successful event. And this time, when we talked about this kind of theme meeting in life sciences, there were other stakeholders as well, but we from RRCAT was very much eager to hold this kind of theme meeting at RRCAT for 2 reasons. One, as I have already told, that to foster collaborative research between RRCAT and other constituent institutes, and those constituent institutes where life sciences are there, they should be able to know what kind of facilities or what kind of life sciences we at RRCAT do. as I told you earlier in other meetings, that many a time RRCAT was not in the map of life sciences as far as in the map of HBNI as far as life sciences is concerned, because there is a small group of life sciences, but this life science group does very different kind of research. That research is biophotonics, and this is a multidisciplinary field. And now I am sure that all the faculties and students who have come here, they have a kind of glimpse of what we do at RRCAT. And many of them have shown their willingness to collaborate with us at RRCAT. And that is a good thing. That is a good thing, and that serves our purpose. And I am very much thankful to our director for agreeing to hold this conference at RRCAT. And I would thank also our Dean Academic of HBNI at RRCAT, Professor Mishra, to really give his full support, and Professor Pal and Professor Dr. Ajit Upadhyay. As far as HBNI Central Office is concerned, I would really am thankful to our Honorable Vice Chancellor, Professor Vasudev Rao, because of him this kind of theme meeting has been successful, and it is the brainchild of none other than him. So he only conceived this idea of theme meetings at different constituent units of HBNI, and he mooted this idea. And everybody in the HBNI Central Office, including Professor P.D. Naik, Dean HBNI, Professor Sunil Ghosh, and other associate deans, they readily agreed. Professor Dilip, so they readily agreed. Dilip Maiti, so and I would say that Professor Sunil Ghosh, I am really, I acknowledge his every support, and at every stage he has given us guidance how to proceed. I'd like to have a big applause for him as well. And Professor Dilip Maiti, who has been here and who has left today, and Professor P.D. Naik, both of them have supported unconditionally, and whenever required, they have given us the kind of guidance. So this is really amazing. I didn't expect, or we at RRCAT didn't expect that kind of support. But one thing I just put on record here, that this HBNI theme meeting, neither HBNI nor BRNS gave any kind of financial support. So there we really got, I mean, when Dr. Nakhe agreed, so our main thing, he was very much worried about where from the funding will come, because he had agreed, because he has keen interest in this kind of theme meeting, and so it has been possible, let me tell you very honestly, by our industry partners who helped unconditionally to hold this event and to make it a successful event. Whatever we wanted, they have provided. Right from our dinner, lunch, everything, each and every penny they have provided. So this is really, they need a big applause. So I would show you the industry partners, and we have, in honor of them, we have kept a small ceremony to honor them. So those who have been here, those who have not left, so they will definitely come, and I would request you to honor them personally. So with that, I conclude here. Thank you very much.
Thank you, sir. Now, I would request our Honorable Director, Dr. Nakhe sir, to please deliver his closing remarks for the ceremony.
Professor Vasudev Rao, Vice Chancellor HBNI, Professor Ghosh, Dr. Satyaram Mishra, Dr. Majumdar, Dr. Paul, Dr. Ajit Upadhyay, dignitaries in the audience, my colleagues, HBNI students and industry participants. Very good afternoon to all of you, and I thank you all for being with us on this validatory function of HBNI theme meeting on life sciences. As mentioned by Dr. Majumdar, this was a brainchild of Vice Chancellor Professor Vasudev Rao, who has been instrumental in introducing new schemes and new ideas to foster collaboration among various institutes of HBNI and interactions among students and faculty members. The discussion meeting on basic sciences was a grand success at BRC, and I attended it, and we were very much eager to have such type of meeting at RRCAT, and I thank Professor Vasudev Rao and Central Office of HBNI for granting this meeting on life sciences to RRCAT. RRCAT, as informed earlier, is well known for its contribution in lasers and accelerators and its applications. But application spectrum definitely covers biophotonics and applications in the area of life sciences. But we had a very niche application area, and we were very much interested to project it in a big way and to have interactions with other institutes so that we can collaborate further. And this is what I appealed during my talk during the inaugural session, that please interact with the faculties here, students here, have exposure to the facilities here, and explore the possibilities that where we can collaborate in future. And I'm sure you must have done all that. And I'm sure, like all good things, although this meeting is coming to the end, but these collaborations and subsequent fruits will be much sweeter, and will have much more useful interactions in future, and will be able to contribute much better in the research and as well as nation building in totality. So I'm sure with these contributions from all in future, will make this meet a successful and role model for other meetings to follow. I take this opportunity, as Dr. Majumdar has said, to thank the industry participants for providing the support, for organizing this meeting in terms of financial support. But I'll go beyond that, and these partners are not only financial supporters, they are participating in incubation activities and taking departmental technologies and propagating them through the innovative ideas added to the technology and coming up with very useful products. So OncoDiagnoscope is one such product, and then Dr. Cure and Care, they're coming with product in the area of biophotonics, and RTK Meditech, they have taken on the this tuberculoscope. So such products they have taken, they have shown confidence in the technology that has been developed within Department of Atomic Energy, and they are coming for technology transfer and incubation activities also. So I'm sure this relationship will be very useful, not only for Department of Atomic Energy, but for HBNI students also, where these students can take summer break or say participate in developing the products based on DA technologies or the research work that they themselves are doing. So this industry participation will be very fruitful even in future also. So with these remarks, I thank once again all of you for being here, and especially Professor Vasudev Rao for his blessings and his personal presence here and taking keen interest in our RRCAT activities. Thank you. Thank you.
Thank you, sir. Now, I would request Dr. Sunil Ghosh, Associate Dean of Life Sciences, HBNI, to come and share his views.
Professor P.R. Vasudev Rao, Vice Chancellor HBNI, Dr. S.V. Nakhe, Director RRCAT, other dignitaries on the dais, dignitaries of the dais, faculties, students, ladies and gentlemen. A very good afternoon to all of you. So now we have reached to the end of this session for scientific discussion, but still it is not end because tomorrow we have another program. Okay, that I think that will be also equally exciting for all of you. And I think all of you will agree that RRCAT has done a commendable job. And I thank Professor P.R. Vasudev Rao for conceptualizing this thing, because he told me in the month of May, Dr. Ghosh, can you arrange the life sciences meeting as early as possible? So that's why we actually made a meeting online, but because of the extreme weather conditions in the May month and then after that rain, so earliest possible was agreed by RRCAT to held in September. So that's why this September date was chosen. So I thank you, sir, very much for that. I thank Dr. Nakhe, Director RRCAT, for his wholehearted support. Without that, it would have not been possible. So conceptualization by Professor Rao and your support. And then finally, it was precisely executed by Majumdar, Professor Majumdar and his team. That's very important. He has done a commendable job in that. Let us give a big applause for him. Thank you very much, Professor Majumdar. And I think all the deans, they are telling that they would like to hold it here in the name of their institute. Okay, so thank you very much, Professor Majumdar, for your leadership, for leading this program. I would like to say that we had a very highly informative, interactive lectures by our faculties and also 3 outside invitees. All the speakers showed keen interest to share their knowledge and also facilities. And they are all open for collaboration. I think they clearly presenting, while presentation only they were telling that they are looking for collaboration. So I think this is a very good platform. I think already there must be some kind of discussion. I found that the students are actually, they have done their job. That's very important. If you see that the students were there for last 2 days, they were standing for 2.5 hours and continuously explaining to people without any interruption. So they have made this interaction meeting very lively. I think all of you have enjoyed. The student, all the students have interacted with all the faculties, and all the students interacted to all students. That's the beauty of this gathering. So I thank them all again for their involvement and making the ecosystem very lively. A fraction of students and faculties have joined physically, but if you see, a large number of participants have joined online. And from what I got the number from my colleague, Professor Maiti, told that more than around 280 people have joined online. I think that's a big number. That's a big number. Okay. So although you are feeling that you are only 100 people here, but you must remember that 3 times of this number of people are actually listening to your lectures. Unfortunately, I don't know whether they could see the poster session. That could have been much better. I think this theme meeting is liked by all of you, everybody. Okay, so I think this will be a kind of model, or we will follow such closely such a model, so that the future theme meetings on life sciences and all other subject-specific theme meetings will try to follow this model, so that I think it will be acceptable by almost all kind of atmosphere. Finally, I thank all of you for active participation in all 3 days. And I wish that all the participants not to miss the tomorrow's morning
marathon. Please join. And after that, the visit to RRCAT lab. So that is my request to all the participants. I think then it will be a much more bigger event then. Okay. And with this, I thank you all again for that. Thank you.
Thank you so much, sir. Now I request Dr. Satyaram Mishra, Dean Academics HBNI RRCAT, for his comments.
Honorable Chief Guest of today's function, Professor Rao, Vice Chancellor of HBNI, Dr. S.V. Nakhe, Director RRCAT, Dr. Ghosh, Associate Dean HBNI, Dr. Majumdar, convener of this event, Dr. C.P. Paul, Dean Student Affairs of HBNI RRCAT, Dr. Ajit Upadhyay, Head of the HBNI activities, dignitaries in the auditorium, students and participants. So today we are here that we are completing this meeting, and all of you would agree with me that the meeting was extremely fruitful. Fruitful in the sense, for doing any productive or research-oriented work, we definitely need two things. One is expertise and other is facility. And now we are exposed to both these things through this meeting. We know where are the expertise and where are the facilities. So now the forum is open for all of us to do a very fruitful research in this direction. And deliberations during the meeting were excellent. I noticed that none of the lecture was gone without any question. At least 4 to 5 questions were asked in each lecture. So that shows the importance of curiosity of the participants. It is not only lecturer, it is participants also. They were very curious, keen to know the things and to know where they are lacking, where they need collaboration. So I think this objective of the meeting was fulfilled in my view. The other thing is that this is also a learning for all of us, that not only in life science, but we should also have such meetings in other areas. Because, you know, once we know each other, who is expert in which area, then the life is easier to collaborate and do the collaborative progress. So for all this, the meeting was conceptually originated from our chief guest, Professor Rao, in May, in the Life Science Discussion HBNI meeting where we all went. So that was his proposal that we should arrange such meetings. So then we thought that we should have this meeting, some subject meeting in RRCAT. We discussed with Director RRCAT, Dr. Nakhe readily agreed for hosting this meeting. So we started from life science. And so I thank him very much for his great enthusiasm to conduct this meeting. He never said that there will be problem. He said, no, we should go ahead for the meeting. And then we thank Professor Rao and his colleagues, Dean HBNI, Professor Nayak, Professor Ghosh, who did a commendable job to coordinate the meeting, plan the meeting, and finally help in organizing the meeting at RRCAT. So that is how this meeting is successful here. And I congratulate Dr. Majumdar for organizing this meeting very efficiently, in my view. And all of you will agree that most of the things were done very meticulously by him and his team. So I also thank Ajit for helping him in a big way. So if you want, you can cheer them. Thank you. And it is very privileged for RRCAT HBNI program here that we have been host for this meeting. Great honor for all of us here. And I hope we can organize such meetings in future also. And we look forward the participation of as many as people possible from other CIs and other institutes. So thank you all. Thank you, everyone.
Thank you so much, sir. Now I request Dr. C.P. Paul, Dean Student Affairs, to say a few words.
Honorable Chief Guest of the Day, Professor P.R. Vasudev Rao, respected Director RRCAT, Dr. S.V. Nakhe, other dignitaries on the dais and off the dais, my senior colleagues, and team meeting participants. So far we had been discussing about advantage of the academia, what we got, what we learned, what we saw. Being the person holding the responsibility of Dean Student Affairs, I was seeing a different angle. The angle I was seeing, whether the students who are coming here, are they gaining anything beyond the academia or this? I was looking into the part, are they having a break from regular research so that they see a different, world, this world in a different format? Are they ready to see the presentation, learn the things with the open mind? And probably to a great extent we could do this, and we achieved to the great success. Another part that I was also looking into it, when we come and go to the outside places, probably at the back of the mind we heard this about this city, we heard this about this city. Did we have a flavor of that in that short span of time? Yes. And I will say that many of you might have seen beautiful campus, but we have at RRCAT, peacock dancing. Many might have seen first time in their life, right? So this is the thing that, brought a different angle to the, added to the theme meeting that we have here. So, and this was only possible because of enthusiasm we have. Yesterday night, I'm just recalling, we all had dinner and we finished by 9:30. Somebody said, why don't we go to Sarafa? And see the enthusiasm people have. After having dinner 9:30, people are ready to go Sarafa and coming at 1:30. So that's sort of enthusiasm we're talking of. So it is not only the organizers, but also the participant reciprocated, and it made the things, and it made it very easy for us to organize meeting. So all these things, we heard a lot, new things here. And being a mechanical engineer, when I see, I was thinking, what will I learn here in bioscience? Nothing. But I saw now things are not that way. Being the people working, being myself working in the mechanical engineering and the material science, I see here also people are doing a lot of things which can be influenced on. Really, it is helpful. So I will say that there can many interdisciplinary talks in this, which will open new door. And definitely, the way things are happening, that day is not far when it will happen. With this, I close my talk. Thank you very much for patient hearing to me.
Thank you so much, sir. So the active participation of PhD scholars from constituent institutes has made this event more relevant and meaningful. With such wonderful participants awaiting the results of poster session, the time is apt to redeem them from their curiosity. So finally, the time for the distribution of best poster certificates. Now I would like to request Professor Majumdar sir to kindly come and conduct this session.
So as Mahima told that this is the session where we will be, or this is a time where we'll be distributing the awards, best poster awards. And as I told you, there are 48 poster presentations, and we have a committee who evaluated those posters. And they had a tough time, and that committee comprises 5 faculties from different CIs. And they have given their decision, and based on that, we will be distributing the awards. And let me tell you that there is no first, second, third prize, and it is very difficult. It is a very difficult task to evaluate, as all of you agree, that all the poster presentations were very, very good and of a very high standard. But for the sake of, this is a kind of judgment, this is a very much personal judgment probably people have taken. So those who will receive the awards, they should not think that they are the best, though they are getting the best poster awards. And those who are not, they should not be disheartened that their work was not noticed. It is not that. It is a kind of, game. Let us have a fun of it. So with that, let me now request Professor Vasudev Rao, please distribute the awards. And I request Professor Dr. S.B. Nakhe, please also join. So I will just read the names one by one, and please come onto the stage. So the first poster for the first Best Poster Award, I would call upon stage Ms. Pradnya K. Pachpati, HBNI BARC, on her work on selective turn on fluorescence sensor for intracellular labile iron pool. Next, I would call upon stage Rupasri Brahma, HBNI SINP. Rupasri, I think she is not there. Next, I will call upon stage Ms. Sucharita Chatterjee, HBNI RRCAT. Next, I would like to call upon stage Elena Bhattacharya, HBNI Niger. Next, I will call upon Ajaya Kumar Sahu, HBNI IMSC. Next, I would call upon stage Miss Nidhi Mehrwal, RRCAT, HBNI RRCAT. Next, I would like to call upon stage Darshan Mehta, HBNI ACTRAC, TMC. Next, I would call upon stage Ms. Priyanka Sengupta, HBNI SINP. She is not there. So SINP students have gone for outing. Next, I would like to call upon stage Ms. Sai Sarvani Mund, HBNI RRCAT. Next, I would like to call upon Naresh Kumar Gudigamala, HBNI Nizar. Next, I would call upon Saurav Chakrabarti, HBNI ACTRAC TMC. Next, I will call upon Ms. Mahima Rastogi, HBNI RRCAT. So that ends the awards. I would thank you, sir. Oh, okay. कहाँ disappear हो गए थे? रूपश्री ब्रह्मा. तुम्हारा दूसरा फ्रेंड कहाँ है? Priyanka. She? I think she doesn't know. So how somebody will know that she has got it? I think so. Please go there. इसको रख लो बाद में. उनको बुला लो. Thank you, sir. So let me tell you that there were 12 best poster awards, and this has been generously sponsored by Research India Bhopal. A big hand to Research India. And they have told that in all HBNI theme meeting on life sciences, they will sponsor this award. So now I will hand over the mic to Nidhi, Mahima.
So with this, the poster session, the award for the poster session ends here. And now I request our director, sir, to kindly felicitate our honorable chief guest for the day, Dr. Professor P.R. Vasudev Rao. So I invite Dr. Majumdar sir again for the distribution of mementos to our industrial sponsors.
So as I told you that this particular theme meeting had not been successful but for the generous funding from our industry partners, and as Dr. Nake already told how they have really come forward to sponsor this event, not only financial support, they are basically related to RRCAT in terms of taking the technology through incubation centers, and they have readily agreed to take or shown the willingness to take the technologies developed at RRCAT, and they are now commercializing it, and they are very much available in the market. And as Dr. Nake already taken the example of OncoDiagnoscope, that is the cancer diagnostic device, screening device, and tuberculoscope, which is a fluorescence microscope, miniaturized fluorescence microscope, doing the same job as high-end fluorescence microscope. And there are other products as well, like Raman Probe and OncoVision. So all the products developed at RRCAT, they have been transferred to industries, particularly from this, I am talking about related to life sciences. And other than that also, non-life science things also they have taken. So now here is a list, as you see in the screen, that Dr. Kioran Kier, New Delhi, Dr. Ms. Research India, Bhopal, PG Tech Private Limited, Indore. They have contributed in a big way. So give a big hand to them. Apart from that, there are several other industry partners who have contributed also in their own way. And among them are SK Traders, Indore, Applied Optical Technologies, Thane, Tragut Solutions, Mumbai, Made in India, New Delhi, Sensil International, Mumbai, Lab India Instrument Private Limited, Mumbai, SLP Solutions, New Delhi, Balaji Cells, Indore, Kasliwal Biomedics, Indore, and RTK Meditate, Indore. So many of them are not, the representatives of many of these companies or industry partners are not present, but I will call upon stage one by one. If they are here, please come on. Their representative should come here. So first, I would like to, I am seeing Dr. Kieran Care, New Delhi. So I would like to call upon stage Mr. Bikas Wahi, proprietor and CEO of Kieran Care, New Delhi, and who has been involved in our incubation center. And I would request Professor Vasudev Rao, please come forward and honor them with a memento. So next is Research India, Bhopal. His proprietor and CEO is Mr. Kuldeep Patel, but he is not here. He has sent his representative. Please come on stage. Thank you. Now I call upon PG Tech Private Limited, Mr. Ashish Tiwari. Mr. Ashish Tiwari, I think he has not been able to make it. Next is SK Traders Indore. Next is Applied Optical Technologies, Thane. Next is Made in India, New Delhi. Okay, I don't think he has been able to make it. Next is Sensil International, Mumbai. I don't think they have left. Lab India Instruments Private Limited, Mumbai. No, not his. SLP Solutions, New Delhi. Balaji Cells, Indore. Kasliwal Biomedics, Indore. He is not there. RTK Meditech, Indore. Mr. Hemant Barma? No. Okay, so they are not here. Thank you, sir. Thank you. Please take your seat. One student has come. So, Ms. Priyanka Sengupta. Thank you, sir. So now I give it to Mahima. So now I will सर को बुलाना है कि तुम भी टाइप ये करूँगा तुम बोलना है ना चीफ गेस्ट को बोलने से पहले
So now is the time to hear from the words of wisdom from the chief guest of honor for today, Professor P.R. Vasudev Rao. But before I invite him to the podium, I request Dr. Majumdar sir to kindly come and introduce him to the audience first.
So I know that Professor Vasudev Rao doesn't need any introduction to the DA family, but still it is my pleasant duty to introduce you, sir. Please. So Professor Vasudev Rao was formerly the director of Indira Gandhi Center for Atomic Research. He is a specialist in the chemistry of nuclear fuel cycle and was responsible for the development of several facilities for R&D on first reactor fuel cycle at IG Card, Kalpakkam. Professor Rao obtained his BSc degree from Madras University and thereafter joined the Department of Atomic Energy in the year 1972 to the prestigious 16th batch of BRC training school. He worked in the radiochemistry division of BRC till August 1978. He obtained his PhD degree in chemistry from University of Bombay in 1979 for his work on chemistry of actinide elements. He moved over to Indira Gandhi Center for Atomic Research, Kalpakkam in 1978 and rose to the position of director, IGCAR. And then he superannuated demitted his office in August 2015. He has over 300 publications in peer-reviewed international journals. Dr. Vasudev Rao has guided 15 students for their PhD degree. He founded the Southern Regional Chapter of IANCAS and through IANCAS organized and taken part in a large number of programs in schools and colleges to educate the students regarding radioactivity and its applications. He also founded the Society for Advancement of Chemical Sciences and Education, that is SACSE, at Kalpakkam, and was its first president. Dr. Rao was selected for the MRSI Medal Lecture in 1998 and the MRSI Senior Superconductivity and Materials Award in 2011. He was also selected for the Silver Medal of the Chemical Research Society of India in 2011. He was given the INS Award in the year 2007 for his outstanding contributions to nuclear fuel cycle. He was one of the vice presidents of MRSI during 2015 to 2018. He was elected as a fellow of INAE, that is Institute of National Academy of Engineers in 2018, and fellow of National Academy of Sciences, India in 2019. With this brief introduction, I would like to invite you, sir, for your lecture.
Thank you, Dr. Majumdar, for a long introduction. It could have been much shorter because everyone here knows me. Anyway, very good evening to all of you. Dr. Nakhe and colleagues from RRCAT, Dr. Majumdar, Dr. Mishra, Dr. Upadhyay, Dr. Pal, my own colleague from Central Office, Mr. Ghosh, and other participants here. A very good evening, and I know that I am standing between you and the cultural program. And in any case, I am not a specialist in life sciences. So I hope you are not expecting anything very sublime from me. But having come here, I thought I will share some random thoughts from my side on this event. And before that, first I would like to congratulate the students for participating in such good numbers and so actively. And I was very happy to hear from everybody about the very, very excellent interactions which happened during the poster session. I believe as the Vice Chancellor that the students and then the faculty, they are the most important part of the university. If we progress, it can only be because of the excellent contributions from students. Faculty alone will not be able to succeed unless students are very good. So I am very happy that we have a good lot of students who are interested, and I wish you all the best in your career. The second point I don't want to forget is to thank Dr. Nakhe and the organizers for inviting me here to this beautiful campus. It has always been a pleasure for me to visit RRCAT. I must mention that after taking over as Vice Chancellor, the first organization I visited was RRCAT. And that not only the campus, the way they focused on development of technologies and transferring them to industry and benefiting the public and doing everything professionally has always very, very impressed me. I must record that in my 5 years of experience at HBNI, the decisions taken in the academic committee, council, or standing academy of deans, etc., were most effectively and quickly implemented by RRCAT. So this is something which I've always acknowledged, and so RRCAT is a very important part of HBNI. Now coming back to this meeting is significant for very many reasons. First of all, I am very happy to find that on the very last session, nearly 80-85% of the original participants are here. That itself shows that the meeting has been very, very interesting to everyone. And I must thank the team at RRCAT and also my colleagues at HBNI for planning this event very meticulously and carrying it out this way. Now coming to the subject itself, subject of the meeting, as I said, I'm not an expert, but I must mention that since every PhD thesis finally passes through me, I do have a look at what the experts have to say about the thesis. The reviewers' comments are all there. Every time I go through the reviewers' comments, and I'm very, very happy to find that, especially in the area of life sciences, of course, it is true for even other subjects as well, but particularly in the area of life sciences, I get very appreciative comments from the reviewers, including the international reviewers, and occasionally statements like, I have not seen such excellent work of international standards. So it's a very nice feeling that across the CIs of HBNI and OCC, the work being carried out is of world standards. I must mention here at the same time, of course, since a number of students are present here, that occasionally I do get comments from the reviewers, the work is excellent, but the communication could have been better. Especially they point out that some English language requires improvement, etc., etc. I will be speaking to students tomorrow. I would like to dwell on this subject a little more, but that doesn't draw away from the excellence of the work that has been carried out by students. So if that is so, then what is there? What is the way forward? What is for me to contribute? There are two things I would like to emphasize. One is that we are all working with taxpayers' money, and Department of Atomic Energy has tradition and in its genes application to society and benefit to society as its important goal. And I would be very happy if work done in life sciences disciplines ultimately gets translated some benefit to the human beings, in addition to, of course, the understanding which develops regarding various things. And it's from that point of view, it's quite significant that this meeting is being carried out at RRCAT. I think it's very appropriate because RRCAT is very well known for taking the technologies to the implementation level with the help of industries. I would think that the recent pandemic brought into sharp focus the important societal impact that can happen from biology research, life science research. Because the development of vaccines in very quick time and the large-scale manufacture, these two things we have to look at together. How they was quite quickly multiplied. It is what enabled mankind to just live through the pandemic and control its effects. And we are very fortunate that several of the HBNI faculty have in fact been engaged in studies related to COVID and other viruses and other diseases, and equally importantly, the understanding, prevention and treatment of cancer. And several other colleagues, I know they are studying, for example, in BRC, the biological effects of radiation, use of radiation in agriculture and so on. All of these have direct benefits for society at large. And I think all these faculty members deserve a great salutation. And my request is that the discussions about taking the work forward to society could happen in these meetings more and more. And that is one of the expectations from this group of meetings. Of course, I must acknowledge there was a statement about financial contribution. I must say that yes, HBNI was not able to contribute to this program. We should discuss the reasons separately. But I can only mention that in the budget for the year 2023-24, we have made provisions for such faculty meetings. So this is one good news. Like any other discipline, if beyond this aspect of collaborations and taking the topics to society, if there can be anything else which can be thought of as a way forward, I would think how to have interdisciplinary or multidisciplinary collaboration. I think that is even more important if we have to make an impact. Like any other discipline, for the fruits of understanding and research in life sciences, if it has to float to the society, I think that interdisciplinary efforts are absolutely essential. It is very appropriate, therefore, that we are holding a meeting here where we have examples of such efforts. We can be proud that RRCAT has developed the oncodiagnoscope, which was mentioned earlier, probably in the meetings, for early diagnosis of cancer. I understand that they have also developed tools for diagnosing tuberculosis in a similar manner, which means that a great combination of strengths in biology, physics, chemistry, electronics, even mechanical engineering, Paul is here, and computer science, all of these subjects, a combination can be a great enabler for development of that products finally about which we can be proud that after we retired, okay, something I made and now it's being used by thousands or millions of people in the country. That kind of satisfaction cannot come from anything, including publications. If you have developed a product, finally you have touched a part of society, I think that's ultimate. And the people in the faculty and the students in life sciences have great opportunity for that compared to any other discipline. Today, every one of us is familiar with the use of biosensors in day-to-day life. without any thinking, whether whatever work you do, whether you are a banker, whether you are an artist, whether we all use biosensors, we know how the subject of biology combined with other subjects, other disciplines like electronics and instrumentation have made a great difference for us. We use, for example, we have commercial continuous glucose monitoring systems. Recently, I have seen that they had started also in India. You just put a tape on your slightly below the skin, you just insert the sensor, and throughout the year, throughout the day, it records the glucose levels in your blood, and you can even download the information on the laptop, and through that you can understand how your food is being assimilated and how your pancreas are functioning, and that can be basis for treatment. This could not have come just from an understanding. It would have come only because group of doctors, group of life science experts, group of instrumentation experts have worked together. Well, we have similarly several other topics where one can understand that today, of course, during COVID, we all understood how pulse oximeters overnight became so important. Well, this came because of a group of people working together. And we had even started getting tools at home to test COVID quickly, rapid antigen tests. This all happened because of interdisciplinary, multidisciplinary collaborations. And I would imagine that in future, we will have meetings where we will try to introduce other disciplines together with life sciences to see what way we can translate the excellence that we have in research and understanding towards real products for the society. I want to take a few other examples. I'm very sorry if all of them are not life sciences, maybe more on medical and health sciences. That is where I understand. Robotic surgery, for example, is another important domain where work is going on even in DAE, in BRC, for example. I know several HBNI students are involved. Only a few days ago, some student explained about his work. Combination of mechanical engineering, instrumentation, and medical and life sciences. Finally, helping people to locate, to arrive at a system by which one can precisely target a particular location in the body and carry out a surgery robotically. And this is something of great boon to society. Similarly, medical prognosis has constantly and significantly benefited from the application of machine learning, deep learning, and bio-inspired computing. I've spoken about this at several places because I have come across very interesting research papers, including one in 2021, which suggests, for example, that in hospitals you have, let's say, in India, among the cancers for women, breast cancer is the most important type of cancer. And people go through several investigations, including mammography, etc. But these papers suggest that artificial neural networks based on thermometry and other sensing methods can very effectively and in early and accurate detection of breast cancer can take place. Even before a mammogram can find out, it is possible for us to locate that this person has a potential to get this cancer. So this comes from a combination of medical and life science expertise and information technology expertise, because artificial neural networks is a topic that is woven into this. Another research topic which I've been passionate about, I don't know whether any of you in the audience I've already spoken to, is very often I speak about is the subject of boron neutron capture therapy. You know, I have seen this actually in a reactor when I have gone to Argentina. That day they said reactor is closed, you cannot visit because the patient is being taken inside the reactor. I was surprised why patient should be taken inside the reactor. So they mentioned that this patient is having certain cancer of the brain. I don't know the medical terminology. What they have done is they made a compound with boron and injected it such that this compound is very selectively absorbed by the tumor. And now, without touching him, without any invasive thing, you just expose him to a source of neutrons. Neutrons go in and then get absorbed by boron and release the alpha particle, and then they kill the tumor cells around. And this is called boron neutron capture therapy. In MIT and several other institutions, this has been developed to quite extent, and even recently there have been papers. In fact, I found that IAEA and just 2 years ago, the International Atomic Energy Agency organized a meeting on this subject, And IAE and Japan's Okayama University have now signed an agreement that provides a 3-year framework for enhanced cooperation in this area. So what does this mean? This means that we need understanding of life sciences, about the tumors, about what compounds they can selectively reach there, what boron compounds can selectively reach there. We need chemistry expertise for synthesizing such compounds in very pure form. We need medical people to be able to really carry out the entire process, and we need expertise in neutron sources and related technology. So you require such a big group of people come together, but they can make a difference to a cancer patient the way no other person can make. So this is why I felt that this is another excellent example of multidisciplinary research, which can easily be done in DAE, because we have all the ingredients required for this, including, I keep saying that we have today in the country through the heavy water board, the possibility of producing highly enriched boron. Boron in the normal boron contains only around 20% of boron 10 isotope, and that is the one which absorbs the neutrons. 80% is boron 11, which is not effective. But the enriched boron produced by heavy water board is 95% in boron 10. And that means that you need to inject into the human body hardly 1/4 or 1/5 of the quantity of boron required for the same effective absorption to take place. So we have that kind of facility. And we have knowledge and technology available at BRC and RRCAT, many other places, about generating neutrons through even transportable sources. You need not take it to reactor. You have today possibilities of making, using accelerators and many other ways to produce neutrons. So all the ingredients required for this therapy are available in the country. And I have been urging at every meeting that this could be one of the important topics. If a group of people across the disciplines come together, it is possible for us to do. I'm just trying to give a few examples just to underline the impact one can make if we gather our strengths in multiple disciplines. And everybody, there is a saying that in DAE, there is no single problem which cannot be attended if a group of people join together, because the strengths are there in all the institutes in some way or the other. And we are very fortunate that in that the constituent institutions and NICER in HBNI have strengths in all these disciplines. And all we need to do is an imaginative choice of the programs and the willingness to cooperate across barriers of disciplines. If you do that, it is quite possible to achieve breakthroughs. So I hope I have conveyed some thoughts from my mind and I have not bored you too much. I am aware that I focus more on health sciences than life science, but that was due to the examples which I chose. But all through my young days and even till today, I have always been fascinated by very silly, perhaps questions, but important questions, like whether we can save lives more if we have, if we chemists and biologists come together and create artificial blood. Shortage of blood is very well known to be an important reason for loss of many lives. And but what I have read quite recently, I think last year there was a paper on this, that even though scientists have not been able to create artificial blood in spite of very large efforts, there have been advances, because now they are discussing that instead of making blood, can we make individual components of the blood artificially, like via plasma or other components, white blood cells or platelets or whatever it is. So bioengineering of blood, I feel, is another very important topic, a multidisciplinary topic for the future. I do not, I'm not the expert to say whether we can do it or we cannot do it. I'm just what has come to my mind, I'm just trying to say. But of course, one important point in this was pointed out, that difficulty in carrying out clinical trials, because we do not know whether there will be willing individuals for getting injection of platelets into their blood vessels. Anyway, I am sure that many such topics would have been discussed during this program. Though I could not join the program personally, I must say that right from the inaugural session, off and on, I was tuning on to the webcast, and I must say that every time I used to connect myself, I found discussion was going on. So this is another very important plus point. I should say that I am very happy that in addition to just lectures, a lot of discussion has taken place, and a lot of ideas for collaborations have come, and this augurs well for all the faculty and for HBNI. And I am sure that this meeting would catalyze further meetings, and I don't see why we cannot hold meetings like this every alternate year at different venues, so that people are able to go and personally visit and appreciate the infrastructure available at different places. Well, I don't want my talk to continue further because we have an exciting program ahead, but I want to conclude by pointing out another important fact which I don't know whether life science experts are aware. The coming year 2023 will be an important year for agricultural sciences. The United Nations has declared 2022 as the International Year of Millets. I'm sure that this recognition of the importance of millets will further spur research and the science behind their beneficial impact on the system and efforts towards enhancing their production, etc., etc. So I think very exciting problems will continue to haunt the life science researchers, and I wish you all the best in your efforts. And finally, I hope that these will succeed in making an impact on the society in some way or other. Thank you very much.
Thank you, sir, for your illuminating words. Now I request our Honorable Director, Dr. S.V. Nakhe, sir, to present our memento to Professor P.R. Vasudeva Rao, sir, as a token of love and respect. Thank you, sir. Now I request Professor Majumdar, sir, to kindly come and take the session ahead.
So that really brings us to the end of this valedictory function. But before I really close it, let me invite faculties from each constituent units first to come on stage and say a few words about their experience of this theme meeting. So let me first start with SINP. SINP, from SINP, anybody? Nobody is there? No faculty is there? They have gone? Traveling? Okay. So then, ACFRAC, maybe Professor Prasanna.
Respected dignitaries on the dais and off the dais. I'll probably speak both from ACTREC, from the point of view of ACTREC, and as the convener of Board of Life Science. So the first thing that came to my mind when the bouquet was given to Professor Rao, they said that sapling will be given. So the magic of RRCAT is such that the sapling turned into a bouquet, and by the time you go, it will become a sapling again. So, and there are also been a confusion of people whether person is from BRC would be from RRCAT, person from ACTREC would be RRCAT. So we decided that we are all RRCAT family members, and this just goes on to say how we are united as a family under HBNI. On a more formal note, I salute Professor Rao for conceiving the idea of bringing us all together. It might sound trivial now, we'll say, this should have been thought about earlier, why didn't we do it? But I think if this step was not taken, collaboration between institutes, which I think will become a distinct possibility after the meeting today, I don't think it would have been possible in any other way. We are all grateful to you for thinking about this, and I think the impact of this will be seen in years to come when we have subsequent meetings, and the duty is upon us to take this forward. I salute you on behalf of ACTREC and part of Life Sciences and all the faculty here. Thank you very much for conceiving this. For me, I cherished high quality science from all the CIs, chirpy and happy students, enthusiastic speakers in such a picturesque surrounding. And like somebody said that some of you would have seen the peacock dancing for the first time. This is my Janma Shabalyam, that I've now seen the peacocks dancing, and I've seen many peacocks around. I've been wanting to see them for a long time, and that to dance. So today my wishes were fulfilled, and thanks to RRCAT for this picturesque place. And so we also, I think in the discussions, we have met with many people, we have discussed ideas, and we have created seeds for collaboration. And as a convener, I think I just wanted to point out 3 things. That a necessity for an incubator was felt by many people. Apart from CI-centric incubators, there should be an incubator for life science, where all the institutes come together with a concrete idea. And me and Abhijit were discussing this, and it should be a more realistic approach, so that in 2 to 3 years, we come up with something that is more concrete that should impact the society. That is one of the agenda. And the second is, I was very fascinated by the facility here for crystallography. And I heard today from today's talk that there are images that are possible by using XRD. And we at the ACTREC are making 3D prints. And one of the things that we discussed with Dr. Sudeep is that how do you quality control? So it's an excellent opportunity for us to collaborate on this for quality control of the 3D printing that we do. These are many things that support the patients. Either it is a support for neck or support for the arm when they are doing surgery and things like that. So it is being 3D printed at ACTREC. So that would be great to see that the quality control is done through these kind of collaborations. And the third was interest. We will capitalize on RRCAT's excellence and expertise in laser, because there is an equipment that we purchased for about 1 crore for quantitative analysis of Western blots. That is something that we detect proteins in small amounts. Quantitation is not possible by the normal methods. And these equipments use lasers. And since we are buying from them, these companies cater so that we'll have to buy the reagents from them, and there are only 1 or 2 lasers. We want more options, it is not possible. So it may be an excellent opportunity to collaborate on a life science equipment. And this, you believe me, this will be used by every institutes across the country, because this is one of the lacunae in trying to understand how the proteins level change and how much it changes with confidence. So it will be a great idea. I'll come up with a write-up for you regarding this. And interdisciplinary collaborations is something we also look forward to. There can be many things that can come up. I think I have also enjoyed being with my colleagues, which hasn't been possible with the administrative duties. So coming here has been fun. I had great time going and eating the street foods. And today again we will travel. And the other important thing to say is about the RRCAT volunteers and their faculties. It's easy to arrange a meeting like this, but to make us closer to you by the way that you have treated us, it's really excellent. We found that you are very open and very friendly and excellent and kind folks. And you can see Professor Mazumdar running here and there to take care of the things, whether it is a mic or a slide operation or anything like that. And another thing that I noticed, photographers, they are trying to help people with the laser pointer or the mic or things like that. It's a phenomenal. I think it just, to me, it's been a magical place with kindness, and I have loved every minute of this. Thank you very much. And we look forward to consecutive meetings in life science discipline and other disciplines. Like Abhijit said, Dil Mange More. Thank you very much.
Thank you, Professor Prasanna, for your kind words about the meeting and RRCAT in general. Anybody else from extract? You want to say something? Please come. Okay, fine. Oh, he wants to come.
So it's just impromptu, so I'm not going to address all of you. We were all part of the same family, and it's this is the first time it's give me a opportunity to feel what the family is. So that's the best part I felt by coming here, and probably tomorrow I will be leaving, and as I'm leaving, I am feeling that I'm part of a very strong family. So I'm going with a lot of hope that wherever we thought that we are constricted, we are having less capacity, we are actually much more powerful than what we can. And I think each of our brain itself is the guide, so we can use this properly, and we will, as the words vibe from our Honorable Vice Chancellor, that the coming and giving back to the society. So I'm sure we'll all together, when we continue this kind of a motion for 2, 3 years, 5 years, I'm sure many ways we have will contribute. So it's a great pleasure to be here. Excellent organization. Right from the beginning, we have been talking, and yeah, some of the talks. It's just incredible interaction that has been part of it, and I think it's very enriching. So it was a great pleasure, and definitely I like to see that such meetings happens again in different other places also, or here. I have no problem. Wherever it is, I'll be there, that's for sure. And thank you so much. And
Thank you, Professor De. From Niger. From Niger? Anybody from Niger? Nobody? Okay. Huh? Huh? Please come. So you have to stand up.
Very good evening, distinguished guest on the dais, off the dais, and already I'm not taking the name, Professor Rao, Professor Majumdar, Mishra, Dr. Nakhe, Professor Ghosh, Professor Paul, and Professor Upadhyay, and many other senior brothers and sisters, all my beloved brothers and sisters. And why I'm telling instead of ladies and gentlemen or these things, because when you stay in a family, you have to tell them as brothers and sisters, and that is our culture, I believe. So we are now in a family. Some of them are elder, some of them are younger, but we are in a family. And I am in Niger for last more than 12 or 13 years. And what I understood when I joined in 2009, that there are different government or autonomous institutes under India government, Ministry of Science and other things. And we belongs to DAE, Department of Atomic Energy. Now I understand that beyond that, there are lots more supports are there, and one of them is HBNI. For last couple of years, we can do research, but the directions, the academic directions, the supports we are getting, as Professor Rao told, he's looking all the PhDs thesis, it's from HBNI. So RRCAT, I believe, is just like NISER, a different extension of family, and HBNI is also a part of that, and really appreciate HBNI. And it's my pleasure that I met lots of my, of course, they are scientists and professors, but I would say that senior brothers who has given me personal feedback for the future. Regarding RRCAT, I believe the other name of RRCAT is success. Why? Because this is not my first time, this is my second time. And that's why I told DAE, where it was a program of DAE without HBN. It was about not an academic program. It was in DAE we have sports and cultural event. And I believe we need a healthy, happy, and physically fit mind and body. Then our first goal should be a good human being, a balanced personality. And DA is also working on that. And I am excited to see Dr. Govind Parchane, and there is other members where I met them in DAE platform. And I really appreciate, as I mentioned, that starting from Peacock, the ecosystem, the Diamond Jubilee Park, the Khush Mahal, or that all these things, and the nicely built indoor facilities of RRCAT, and the food, of course, and the people. It's really excellent, and that's why I told the other name of RRCAT is success and happiness. And we were quite excited in the totally different platform. There was no science, because we should have a very good professional life as well as a passionate life. So this gives us balance for a future healthy life and mind. And regarding the program and theme, there are lots of senior brothers and sisters, and also quite younger brothers and sisters. Probably 40 years ago or 60 years ago, the other name of life science, although it is different, it was biology. And we say now biological sciences. What was that? That was zoology and botany, right? But nowadays, the biological sciences and life sciences is totally changed and transformed. And it is the other name of life science is interdisciplinary science, where hardcore physics and mathematics are implied. You cannot tell that this is the classifications of animal kingdom. This is elephant, this is amoeba. It's not that life science what you used to see, or probably my senior brothers used to read. Now life science has changed. Why? It's very, very simple. It is the necessity. But Professor Rao mentioned that in the recent scenario for that pandemic viral infection, COVID-19, we understood that how much integration is required. And vaccine, since I work in the field of immunology, and probably we are from NISER, we have dual duties, teaching as well as research. And Professor Rao is well versed about that, and that's why it's a great platform to see him and his directions. That vaccine means in biological terms, or more specifically or especially for immunological terms, is generating a good memory. What do we understand by memory, and what do you understand by vaccine? Vaccine means to activate the adaptive immune cells, which has the capacity to generate immunological memory. This is the simple definition or explanation. But for this simple requirement, we need not only biologists or immunologists, but beyond that, an interdisciplinary research. And I am really feel proud to be an Indian that we made the COVID vaccine and which is working. There was lots of question whether it works, side effect. We understood with the help of modern life sciences, modern medical sciences, and immunology also, that it is not about resistance against the infection, but to resist the fatality, right? And this happened with memory. So in a lighter mood, I like to say that we all got vaccination in RRCAT, that what is the meaning of life, a good memory we have received in RRCAT. Thank you very much. With that, I am really grateful for Shobhanda, Dr. Shobhan Mojumdar. In Bengali we say Dada. So I met lots of great Dada here, and it's a really privilege, and also my sisters, that who has really behaved like a core family member. Thank you very much.
Thank you, Subhashish. Any students? Students, anybody? Yeah, please come.
A very warm good evening to Honorable Vice Chancellor, sir, Honorable Director, sir, to all the esteemed dignitaries on the dais and off the dais, my batchmates from HBNI, and all my junior and senior PhD scholars. A very warm good evening. It was indeed an enriching experience to be a part of this HBNI theme meet, which was based on life sciences. We got to hear the research works of all the eminent life scientists of India. It was truly an enriching experience on that aspect. Apart from that, we had poster session. On that, we received valuable inputs from all of them, which will definitely be a value addition to our research work. So I look forward, basically we all look forward for such theme meetings in the future. And I'm extremely grateful to all the organizers of this theme meeting for giving all of us an opportunity to experience and to be a part of such a great event. Thank you so much.
Anybody else? Please stand up. Anybody else? So if there are no, then let us proceed ahead. And I call upon Mahima to conclude the session.
Thank you, sir. Now, I would like to request Dr. Ajit Upadhyay, sir, to kindly come and give the vote of thanks.
Good evening, everybody. It's my pleasant duty to propose vote of thanks. First of all, I would like to thank Professor P.R. Vasudev Rao, Vice Chancellor HBNI, that he has agreed to grace this occasion. We are extremely grateful to you, sir, for sparing time from your busy schedule. In fact, the idea itself was your brainchild, and I hope that we did justice to the idea. Dr. S.V. Nake, Director RRCAT and patron of this theme meeting, deserves special thanks. It is due to his keen interest and constant support and guidance that the meeting is a success. He has spared so much time for this theme meeting and has been instrumental in guiding us from the planning stage itself. In fact, he has participated in, I think, 2 or 3 VC meetings also. Professor P.D. Naik, Professor Sunil Ghosh, Professor H. Pal, and Professor Maiti, all these faculty from HBNI Central Office deserve special thanks for their kind support, help in meticulous planning of event at every stage. I thank all of you, sirs. You have been part of planning and implementation too. We thank Professor S.R. Mishra, Dean HBNI RRCAT, and Professor C.P. Pal, Dean Student Affairs, for their constant support. Professor S.K. Majumdar, Head Laser Biomedical Application Division and convener of this meeting, has been instrumental in meticulously planning and execution of every single aspect of this event. We are grateful to you for your constant support and guidance. All the volunteers who have worked tirelessly to make this program success, they deserve a special thank. I would like to cite a few names here, mention a few names. Dr. Hemant Krishna, Sri Vijay Kumar, Sri Rajesh Patidar, and their teams need special mention for working day and night during this organization. I thank all the participants, the students and the faculty from various CIEs who have taken out time to come to this place, which is not directly connected by train to most of the CIE institutes. Last but not the least, I would like to thank all the people who have directly or indirectly supported in making this event success. Thank you all.
Thank you, sir. Now, I request everyone to kindly stand on their places for the national anthem. जन गण मन अधिनायक जय हे भाग्यत भाग्य विधाता पंजाब सिंध गुजरात मराठा द्राविड़ उत्कल बंग विंध्य हिमाचल यमुना गंगा उच्छल जलधि तरंग तब शुभ नामे जागे तब शुभ आशीष मांगे गाहे तब जय गाथा जन गण मंगलदायक जय हे भारत भाग्य विधाता जय हे जय हे जय हे जय 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 हे।
So we have a cultural evening within 15 minutes because it is already session has been delayed. So please assemble here within 15 minutes. And today we'll have a troupe by one famous artist of Indore. He's a flute. He will perform flute recital with his troupe, and there will be vocal also. So please make yourself available here. Okay, please participate. Thank you.